A
This
very good
conference
morning,
will now be recorded.
noon, or evening to one and all present here virtually with me today. Let me introduce myself. My name is Jessica Reeves, Conference Secretariat at Coalesce Research Group LLC, and I will be available for any of your queries and questions throughout your webinar to help you out technically and organize the webinar successfully. For the next few minutes, please allow me to give a brief description about our organization, Coalesce Research Group, and also for the further instructions. Coalesce Research Group LLC is very delighted to welcome you all the speakers, supporting associations, students, and delegates present virtually with us today. With the aim of mark the scientific excellence by facilitation conversations, sharing knowledge, Coalesce Research Group follows through the objective of setting standards in delivering excellence in the research among the researchers, academicians, and industries, promulgating the research through conducting events and online webinars from various fields of science, especially from the stream of medicine, science, engineering, and technology. Within a short duration, we have succeeded in creating a platform for developing a strong professional network with the people across the world from their respective fields that they haven't connected in a while. As a global scientific event organizer, we plan and carry out international conferences, meetings, seminars, symposiums, and workshops, providing the best delegate experience, excellent networking opportunities, conversations leading to innovations, learning new opportunities, lasting working relationship, and professionalization with the professionals around the global network. Just recently, we have started our own platform for open access peer-reviewed scholarly literature, where all the content is freely available without any charge to the reader. We aim to share the new recent advances through scientific publications, which will help in minimizing the borders and contributing the use of research works of eminent authors, scientists, researchers, research scholars, students, etc., across the broad spectrum of medical, clinical, pharma, and engineering scientific disciplines and its subspecialties to promote the ideas which will guide the development and facilitate the use of scientific information and ensure its quality, and also will be connecting link between researchers and scientific community. We are committed in ensuring that our journals are disseminated worldwide so that the researchers are able to access our content wherever and whenever they need it. As being very a few new journals, we have coined up some interesting journals and have also formed the editorial board, and we will be very glad if any of our today's attendees, speakers, students, or delegates could extend your support by checking our journals and will be interested in publishing an inaugural issue at any of the journals according to your area of expertise. We will also send an email with all the details about the journal after the commencement of the webinar. Do take your time to check the website. Finally, I would like to thank the attendees and participants of today's webinar and hope you will enjoy the webinar and gain knowledge from the sessions today. We also hope your participation will contribute to our new research findings and that will lead to new collaborations and research initiatives. Before we start with the sessions, I guess some of you will be the new to the GoToMeetings app. Let me just explain you the important tools required for the presentation. On the GoToMeetings app or web page, you can find 4 icons at the bottom of your center screen. That is mic, camera, screen, and end. Mic, click on that to mute or unmute yourself. I request you all to stay muted while your speakers are presenting their research work so that it does not cause any disturbance to them. Camera, you can use this button to share or unshare the web camera. If you share the option to share the webcam, you can do it, or it is completely your choice. Screen, you can use this button to share your screen. This is the main part of the webinar. During your presentation, after I call your name from my side, I will be making you the presenter, and only I give the permission you will be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. And this is the 4th icon where you can use this to leave this session. On the top center, you can use the options to change your display settings. At the top right corner, you will have 3 icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For the settings, you can use this button to check for the settings of your speakers and microphone. So as mentioned, kindly remain muted during their presentation so that the presenters don't get disturbed. And for any questions, you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you to please join us a brief introduction about yourself before starting the session so that we can know about you virtually. And now let's move in your next sessions. And the first presenter is Dr. Katsuki Hirakawa from Institute of Health and Life Sciences, Japan. But okay, I think he is not joined till now. So I request Dr. Rosanin Sutping Gray from Mahidol University, Thailand, will present the first speech of today. Uh,
Yes, I can.
I'm sorry, Dr. Rosani. Dr. Katsuki has joined now, so we can further proceed
Okay.
with the first presentation. Hello, Dr. Katsuki. Am I audible to you, Dr. Katsuki? Hello, Dr. Katsuki. Your voice is not audible.
Hello,
Yeah, hello, Dr. Katsuki.
I'm ready to speak.
Yeah, thank you, Dr. Katsuki. It was a very odd time for you, but we will request to start your presentation. I am going to make you the presenter. You can start your presentation.
Okay, screen.
Yeah, it's
Oh,
done. I am
how
making you the presenter. You can share your screen and start your presentation.
yes, I
Actually,
understand, but
your voice is not
for
audible,
some
doctor.
reason it doesn't work. Just a moment. Is this okay?
Yeah, can you do a volume high of your speakers? Like it's not audible. Yeah, it is visible. You can
How
start
do
your
I?
presentation.
Okay. Can you hear me?
Yeah, it is
Can
audible.
you hear me?
Yeah, it's doctor, it's
Can you hear
audible.
me? How to volume up? Just a moment. Is this okay? Hear me?
Yeah, it is okay, doctor.
Then let's go back to my screen. Is it okay?
Yeah, it
Can I
is okay,
start my
doctor.
talk?
Yeah, you can start.
Okay. Then I am Hirokawa from Tokyo. Can you hear me?
Yeah, it is audible,
I'm
doctor.
from,
Please
okay,
show us the presentation from your screen.
slow network. Uh, can you see my slide?
Yeah, no, the slide is not visible. Please click on presentation.
This is the first time to,
Okay, I
uh,
will be I will assist you, doctor. No problem. Please minimize this button, the screen. Yeah, it's perfect, doctor. Please start your presentation.
okay. Uh, Today, uh, this first slide show no conflict of interest. In this webinar, I would like to talk about quantitative assessment of immunological level in human aging and the trial of immunological restoration by improvement of lifestyle. Purpose of this talk first is introduction of quantitative assessment of immunological level in human. The second is age-related change of human immunity by this method. The third is assessment of immunological restoration by this method. Before starting my talk, I would like to explain what is immunity in the first place, because many of attendants or listeners in this webinar may not be familiar with immunity. As shown in this slide, immune system plays an important role in stopping the occurrence of cancer and infection. And recent data have shown that the immune system is related with the development of atherosclerosis. Major problem is that such an important immune system peaks at adolescence and start to decline thereafter with age. The level decline at 50% of the peak level at 40 years old, and as low as 10% at the age of 70. But the rate or slope of the decline, I mean immunological decline, is variable and different by individual. But for the maintenance of our health, we should know the level of immunological function of our cells. But so far, there is no credible method to assess the immunological level. Therefore, we have developed the method to assess the immunological level using peripheral blood of human. We employ flow cytometry and measured number of T cell and its population, B cell, NK cell, and this slide shows the number of T cell and its subpopulation, CD4 positive, CD8 positive T cell ratio, naive T cell, naive memory T cell ratio, number of CD8 positive, CD28 positive T cell, and B cell, and K cell. The last is T cell proliferation index, TCPI. And then, as you can see, data are variable, but it is difficult for us to assess the immunological level at a glance. So, at first, we have made basic data from 2,000 people of various age. Data from each cell population roughly scattered in normal distribution. Here we can separate the data in 3 groups and gave you scores. Score 1 is lower 10%. Score 2 is middle 30%. And the score 3 is top 60% of each immunological level. And using this scoring method, 8 different kinds of immunological data are given scores and then grouped and expressed as a numeral as shown in this slide. In this case, 8 different kinds of immunological data values are expressed as 60/24, as a group. We have named it immunological score. This immunological score is separated into 5 groups, as shown in this slide. In this case, immunological score 16 can be referred to as immunological grade 2. We call it the warning zone. Using this method, immunological level is understandable even those who are not familiar with immune system. We further developed much easier expression of immunological level, just expressing as age. One is immunological age, which is calculated by the level of T cell proliferative index. We call it TCPI. This TCPI declines with age, and it is possible to calculate age using this formula in the bottom. The other is T lymphocyte age, and which is calculated by the number of CD8 positive, CD28 positive T cell. The number of this T cell subpopulation decline with age, and it is possible to calculate age by using formula in the bottom. Once again, one can compare these immunological or T lymphocyte age with his or her chronological age. If the calculated age is higher than chronological age, he or she has noticed that his or her immunological level may be lower than average. As you can see in this slide, distribution of this data is variable. This means that those calculated age are most not definite and just show tendency. But we have definite immunological data indicating actual situation. This slide shows a good correlation between immunological age and T lymphocyte age. And this slide shows next one is this one. These sample of the quantitative assessment of immunological level in 2 people. Left is Mr. Taro, 69 years old, and 8 different immunological levels are shown in radar graph in the bottom. The immunological year is 51 to 68, and T lymphocyte age is 46 to 48 years. Both are younger than his chronological age, 69, so meaning that his immunological level is higher than average. The light is Mrs. Hanako, Ms. Hanako, 61 years old, and the radar graph shows that slightly distorted type. Immunological age is 72 and 75. T lymphocyte age is 63 to 64. This is higher than 61, meaning that her immunological level is lower than average. So using this method, we published several papers. This slide shows the gender difference, male-female difference. The rate of aging is slower in female as compared with male. And we also published several papers for the assessment of effectiveness of various supplements. But here I don't show you for the time limit. And this slide shows the follow-up examination in 2 cancer patients. One is this one is survivor after surgery. Immunological score is maintained in relatively high level. The other is unhappy case who finally died after surgery. Immunological score is gradually declining. And actually, lung metastasis happened, and finally, bone metastasis also happened. So this is a good measure for prognosis of cancer patients. For the last 10 years, we assessed about 18,000 cases. Healthy people are more than 60 and 6,000, and the disease people other than cancer are over 5,000, and cancer patients are about 5,000. As I told you in the former slide, immunological level is separated into 5 grades. By grade 1, 2, 3, 4, 5. And when you look at the healthy people are in this slide blue column. The top is grade 3, and the second peak is observed in grade 4. And but when you look at disease people like this disease red one, peak is grade 3, but the second peak is grade 2, the lower immunological level. And the same thing can be said about cancer patients. Top is grade 3, but second is also grade 2, higher than healthy people. That means these disease people are immunologically lower as compared with healthy people. This slide shows the decline of immunological vigor in cancer patients. The left side shows distribution of healthy people and cancer patients in terms of immunological score. Reds are cancer patients, which are distributed to lower region as compared with control, healthy people, black dot. The right graph is the number of healthy people, cancer patients viewed from the immunological grade. You can see that immunological grade is higher in healthy people, this black line, as compared with red dotted line. The last part of my talk is restoration of immunological level or vigor. This slide shows the 7 methods for immunological restoration. First is improvement of lifestyle. And the second is improvement of nutrition. And the third is antioxidants. And antioxidant substances frequently appear on TV commercial message, although their effectiveness is doubtful. Kanpo hozai is Japanese herbal medicine. This is sometimes effective, but effectiveness is dependent on people. Bone marrow transplantation is sometimes employed for the treatment of leukemia. The last one is the transfusion of self-lymphocyte activated and proliferate in vitro. This slide shows one example of infusion of activated T cell T lymphocyte. Gray column are labeled after the infusion of activated T cell. You can see the significant recovery of T cell number and the T cell proliferative index. This slide shows one example. This slide shows one example. At present, I am now interested in how improvement of lifestyle can affect immunological vigor. So for this purpose, I ask the people to answer questionnaire regarding his or her lifestyle. As shown in this slide, lifestyle by questionnaire, physical condition, 8 questions, dietary habits, 8 questions, physical exercise, 8 questions, sleep, 8 questions, tobacco and alcohol, 8 questions, stress related to mind. But this is to check the individual people realize that there are items that should be improved in their lifestyle. This is one example. In 53 years old female, she has a lot of stress in her family environment. She used to enjoy theater going, but stopped it because of family trouble. So I advised her to go again to theater, and 6 months later, her immunological level restored to some extent. Next is the case of 66 years old male. He is a typical Japanese hard worker as a lawyer. He is enjoying his business, but his business is too hard physically and psychologically. So I used Kampo Hozai for him, and after several months, this score go up to 21 plus 24, and finally full recovery. So this is a conclusion of my talk. First, I introduce a quantitative assessment of immunological level. This method has shown that aging of immunity in human and sexual difference. This method has shown that immunological level is decreased in cancer patients. Normal distribution of immunological level in both healthy and diseased people with different patterns. This method is useful for prognosis of a patient suffering of many diseases. Thank you for your attention. Okay.
Thank you, Dr. Katsuki. It was a nice presentation. Is anyone having any questions?
No questions?
Okay, I think there are no questions. So thank you, Doctor. Thank you very much, Dr. Katsuki. So now we can move forward for the next presentation. It is
Okay.
from Dr. Rosarin Sutipong from Gray from Mahidol University, Thailand. Hello, Dr. Rosarin. Are you ready for to give the presentation? Hello, Dr. Rosarin. Am I audible to you, Dr. Rosarin?
Hello, can you hear me?
Yeah, it's audible, doctor. So are you ready
Okay.
to give the presentation?
Yes, I do.
Yeah. So I'm going to make you the presenter. You can share your screen and start your presentation.
I will try.
It's done. You can click on the button share screen.
I click the PowerPoint presentation, and what should I do next?
Yeah, in the screen of go to meetings, you will be having a 4 icons like mic, camera, screen, and leave. Please click on screen button. Yeah, it's done, doctor. You can start your presentation.
Okay, thank you. Good evening from Thailand. I'm Rosalind Gray from Institute for Population and Social Research, Mahidol University. First of all, I would like to thank the organizer for inviting me to speak on the topic, the influence of co-residential and non-co-residential living arrangements on sufficient fruit and vegetable consumption in the aging population in Thailand. My talk will be based on our paper recently published in BMC Geriatrics. First, I would like to talk about older persons in Thailand. We refer to population age 60 and over. You can see that Thailand ranks the eighth in Asia in terms of percentage of population age 60 and over. Our study focuses on living arrangements in Thailand. Our traditional living arrangement, which is living with at least one child, the top line. You can see that this trend is declining due to socio-economic and cultural changes and fertility decline. While the trends of living with only spouse, the orange one here, and living alone are increasing. An emerging living arrangement is skipped generation household, the yellow one, meaning that grandparents living with grandchildren in the absence of grandchildren's parents. This type is mainly due to the migration of young adults to work somewhere else. Since the study on the association between living arrangements and, I will say, FV intake among older people is rare, thus the objective of our study is to investigate the association between living arrangements and FV consumption among older persons in Thailand. The data is from monitoring survey on fruit and vegetable consumption among Thai population, wave 1, in the year 2018. It is a national representative sample survey. We extracted data on 2,048 persons aged 60 years or older. Binary logistic regression analysis was used to investigate the association between independent variables and fruit vegetable intake. This is the framework of our study. Fruit vegetable intake was the outcome variable. The sufficient FV intake was based on WHO recommendation, which is equal or more than 400 grams per day. For the measurement of fruit vegetable intake, we follow the guidelines of Bureau of Nutrition, Department of Health, Ministry of Public Health, Thailand. The focus independent variable is co-residential and non-co-residential living arrangement, controlling for socio-demographic health characteristics, knowledge of fruit vegetable intake, exposure to community campaigns on FV and household size. I select some important findings for your interest as follows. As a whole, only 32% of Thai older adults consume sufficient FV. Females had a higher percent than males. The oldest all consume least fruit and vegetable. Based on our sample, more than half of older adults live with at least one child. Only 6% live alone. The percent of older adults living in larger households consume sufficient fruit vegetable was lower than those living in smaller households. Those living alone consume sufficient fruit and vegetable the least. Let's see the results from binary logistic regression. Our dependent variable is insufficient FV intake versus sufficient FV intake. The highest odds ratio was living with at least one child, while the lowest odds ratio was living alone. Compared to living alone, the older adults from co-residential households were more likely to have sufficient fruit vegetable consumption. I am explaining the possible reasons for our findings. Living with at least one child had a strongest association. This may be due to the bilateral family system in Thailand, in which adult children, particularly daughters, are expected to provide support, including meal preparation to older adults' parents. Compared to living alone, the older adults from skip generation households were more likely to have sufficient fruit vegetable consumption. This may be explained by the role enhancement theory, in which older adults feel pride in returning the role of primary caregiver and shaper of a young child, giving the enhanced meaning to their life. These older adults may give high priority to a healthy lifestyle, including eating fruit and vegetable. Living with older adult spouse was more likely to have sufficient FV consumption. This may be explained that a couple can provide mutual social support and care, including proper dietary intake. In conclusion, we could say that co-residents can reinforce healthy lifestyle practices and better monitoring of eating behaviors, and thus contribute to increased fruit vegetable intake. For gender, females have a higher chance of sufficient FB intake than males. For age, the association was not statistically significant. The lowest odds ratio for household size, the lowest odds ratio was household size more than 5. Older adults living in large households, that is 5 or more persons, were less likely to have sufficient fruit vegetable intake compared to those living in smaller households. This may be explained by increasing household size leads to increased food expenditure and ultimately lower dietary quantity, quality, and diversity. We would suggest it is therefore important for policymakers to implement interventions to increase access to low-cost, high-quality fruit vegetable through such approaches as self-help, fruit vegetable kitchen gardening. Our study that you can see below the slide, found a positive association between home gardening and fruit vegetable intake. Females have a higher chance of sufficient fruit vegetable intake than males. This might be due to a greater sense of responsibility for own health among women than men, such as being more active seekers of health-related information and being more attentive to consumer products that affect their health. Older men may adhere to masculine norms, such as self-reliance, stoicism, and primacy of work, and thus may view fruit vegetable eating as less masculine. We would suggest enhancing the woman's role in the household. This should have a positive effect on eating behavior of the whole family, supporting women's income, enhancing a woman's food-related knowledge and skills, providing technology for food preparation, and encouraging social networking with other women who are the nutritional leaders of the household. How about men? Men should also be encouraged to be role models for the younger generation in helping with meal preparation and consuming ample quantities, quality, and diversity of fruit and vegetables. I come to the last slide. In conclusion, the older adults who live alone, as well as those living in a large households, are at particular risk of inadequate fruit vegetable intake and require special attention. Thus, the policy should take into account older adults' living arrangements, community context, that is exposure to fruit vegetable campaign, level of fruit vegetable knowledge, and socio-demographic characteristics such as gender, education, occupation. For the association between the other variables that I did not present here and fruit vegetable consumption, you can find them from our paper mentioned at the beginning of my presentation. Thank you for your attention.
Thank you, Dr. Rosarin. It was a nice presentation and was very informative. Is there anyone having any questions?
Hello? Hello?
Yeah, hello.
How are we supposed to ask the question at the end? We have to unmute our mic and speak?
Yeah, you have to unmute your mic and you can ask the questions. Dr. Rosarin will answer the questions.
Okay, okay. No, I just had a general inquiry how to ask questions.
Okay, okay, doctor. I think there are no questions. Thank you very much, Dr. Rosarin.
Thank
Now we
you.
can, yeah, thank you. Now we can move towards the next presentation. And it is from Dr. Myra van Wijs from Radboud University Medical Center, Netherlands. Hello, Dr. Myra. Are you ready to give the presentation? Sorry, Dr. Myra, your voice is not audible.
Yes.
Yeah,
Can
it's
you hear me
perfect.
now?
Yeah, it's audible. So I'm going to
Okay.
make you the presenter. You can share your screen and start your presentation.
Okay, thank you.
Yeah, it's done. So you can click on button screen and share your screen.
Mm, this one. Button screen, which this one?
Yeah, you will be having 4 buttons at the bottom of your screen.
Yes, I have. I think I have it now, and now I can share my screen now.
You know it's not visible, doctor. Is it asking for any permissions?
I need to present. Please choose your. What do I have to do here? Share,
Yeah, share.
and then
Yeah, it's perfect. It's visible now.
yes, I'm visible.
Yeah.
But I want to share my my presentation.
Yeah. Open the PPT which you have prepared for your presentation.
Yes.
Yeah, this one. Yeah. Perfect,
Okay.
doctor. Yeah.
Okay, I'm in now.
Yeah, you can start your presentation.
Thank you very much.
Thank you.
Thank you very much for inviting me for this webinar. My name is Mira van Ooy and I'm a medical sociologist by training. And I'm going to share with you my thoughts on bridging the divide between biomedical and psychosocial approaches in dementia research and the key role of social health in it. And I owe this invitation to our article on this topic, and the article has been written by the Interdem group as a kind of manifesto to set a research agenda for psychosocial research and for bridging our research to biomedical research. And here we are. And we are dedicated to psychosocial interventions in dementia. And we try to cover the whole field, and therefore we make task forces, task force on social health, on methodology, and on prevention. And prevention, that will be the topic of my presentation, and on migration. And we also have an MOU with Alzheimer Europe, meaning that we are the scientific partner of Alzheimer Europe, and we are engaged in public patient involvement. We think that the voice of people of dementia is crucial also in dementia research. We wrote a lot of articles and we wrote several books, and this book is very important about psychosocial interventions, which has been studied and described. And some of them I mention here: behavioral interventions, occupational therapy, counseling at home, meeting centers, assistive technology, dancing, music. These are all potential interventions in dementia. So this is about our work of Interdem. For those who are not familiar with dementia, I give a very simple definition. Dementia is a decline from previous competences and previous attained cognitive level, affecting, I will close something that I can, oh, sorry, cancel. Now I spoil it. Can you still see me? Okay, I'm back. Okay, and dementia globally, 47 million people with dementia around the globe in 2015, and this number is projected to triple in 2050. And I will come back to this later because there is some hope. I will first explain something on social health. Well, social health is the social domain of health. And the concept is in place already since 1946, but not much has been done with it, and especially not in dementia. And it's a relational concept. So it relates to the person, the individual, and the potential of the individual to participate and the potential of the environment to influence the individual. This is very globally, and it's important. It's in place all over life, from being a little child to an old person, and in all health conditions. And roughly, it's about and when you look at the individual, it's about the capacity to fulfill potential. So to fulfill what you can do and your obligations. So the social demands. It's about all kind of conditions. So you can be socially healthy when you have a physical condition, and it's about the capacity of the person to participate in social life. And with regard to the environment, it's about the structure of the environment, whether it's you are living in family structure, you are living alone, and about functions, whether you get emotional support, whether you give emotional support, whether you give practical support. And here are some markers. We call these social characteristics markers. And social health is an umbrella concept. So what we want to do now is to gather all these social markers under the umbrella of social health, so that when you do epidemiological research, you can do something with social health as one umbrella marker. And if you look at some examples, for instance, what is important, the contact frequency, whether your social network is consisted of friends or neighbors or only family, and the perceived relationship quality is important, social support is important, and social engagement. And so in epidemiological research, practically the only marker referring to the individual is social engagement, which is an important one, but it is also the others are important. And this talk is about building bridges. Well, if you look at dementia, it's too narrow if you only look at the physical condition. We have to do better. We have to go into interdisciplinary research. And social health can be a bridging concept. And this might offer a new perspective on prevention and also on living with dementia. I here have a picture of a bridge. That's the bridge in my town. And my town is Arnhem, and this is the bridge too far from World War II. And so far, has been done little interdisciplinary dementia research, at least not interdisciplinary, making the bridge between brain sciences and social sciences. And there are very important barriers. And one barrier is the parochial attitude of us, of researchers who only focus on their own topic. And that is loading the site on the whole picture. And this is what we need to move forward in dementia research. Without that, we don't move. And these comments, these barriers came not from us, it came from the artists who make this, who makes a sculpture brainstorm, and he tried to present these very barriers in his sculpture. And why is it so important that we build these bridges? Because we have to learn much more on mechanistic pathway. And there are no effective biomedical treatments so far. And there are no effective psychosocial treatments so far also. And there is a growing recognition of dementia as a multifactorial syndrome. So if you recognize that it's a multifactorial syndrome, you have to bridge these barriers. And we think social health is a good bridging concept. And I will explain you why we have such a good reasons to do this. There has been found a discordance between neuropathology and ultimate clinical outcomes, meaning that there is room for other markers than biomedical markers. And there is a declining incident rate. That's one of the newest findings. And these declining incidence rate has been found in Europe and in America and not in other parts of the world. So that's also something very intriguing. And there is epidemiological evidence on relationship on health and between health and cognition. So if you want to study something, you need a hypothesis. And our hypothesis is that social health can act as a driver for stimulating and using cognitive reserves through active facilitation and utilization of the individual's social capacities and those of the immediate social environment. I explained something on social health. I will explain something now on cognitive health. Cognitive health, that's something that's really a hopeful concept. It is the ability to actively withstand neuropathology by using compensatory cognitive approaches. And this is crucial. This is meaning that we can do something, that our brain is flexible and we only use a part of it, and we can make bypasses like in this traffic picture. So this is what we try to do. And well, if social health is so important for the brain, and if social health is so important for health, then the measures to fight pandemics and to fight other outbursts, they might be very negative for our health. And luckily, research has been done on previous outbreaks, and many of them in China. And it has been found that quarantine has a negative association with cognitive functioning, especially the way the quarantine is. If it is very close, if you have no freedom, it's worse. It has a negative influence on psychological health, on the immune system, and on social health. So that's where we were. And then we had the major opportunity to get a funding of the European Joint Program Neurodegenerative Diseases to do a study on this, to set up a study, especially with this purpose. And our ambition is to influence dementia process and quality of life. And this project aims to unravel the interplay between social health and biological and psychological factors on the trajectory from cognitive healthy through dementia. And what we did in this project, we did several literature studies, and that's a good start. And we found that there is evidence on, actually my screen is blocked by by the information of the webinar. That there is evidence of the relation between social health and cognition. And the protective factors are education, a good social network, leisure activities, and a socially integrated lifestyle, the frequency of contact. And I will come back to this later. The risk factors are poor social health. And the conclusion from Laura Fatiglioni, and she did a lot of research on this topic, is that stimulating psychosocial and lifestyle factors is protective against dementia. We also found, and we did a study on it, that, well, maybe the combination is more important than the single factors. And that's we found. We found that social frailty or social exclusion, including social isolation, financial deprivation, living alone, and lacking basic allowances, is very negative. So, and you might see it's not only contact between people, it's also about financial deprivation, being deprived, and being deprived from basic social rights. So I now present only the relational aspect, but the relational aspect on a very small level, but the relational aspect are there also on a societal level. So I will want to stress this. And then there is a combination of factors that are beneficial, that stimulate good cognitive function. And you see, it's the counterpart. It's a high educational level, high occupational complexity, and participating in social activities. Social network characteristics are important, like being married and network with friends. Note that networks, combined networks, they are much more beneficial than only family networks. And group participation and work engagement, these are all important factors. And it has been found that the cumulative score of these various showed a dose-response relationship, and those who scored highest were 46% less inclined to develop dementia compared with those with the lowest scores. So this means that these social health factors are important, at least. And then we were lucky again. We had another very important and huge grant that allowed us to study the factors in men and mice. And I'm not going to talk about our mice research today, but I'm going to talk about the research from the Rotterdam data, and that's a large epidemiological database. And in this study, we were able to study brain markers and psychosocial health markers. And our hypothesis in this study was that social health affects brain structure and consequently influences social functioning. And we had only few social health markers: loneliness, perceived support, and marital status. And we had much more brain structure markers because it's a very strong department in Rotterdam. Magnetic resonance imaging of the brain to obtain volumetrics, cerebral small vessel disease markers, and microstructural integrity. I'm not going to bother you with all these brain markers, but the key thing and the key finding is that social health is associated with structural brain changes in older adults. And this is a very important finding, and it's about loneliness, and it's about better perceived social support, and it's about being married. Being married has a beneficial effect. And there is not only research on mechanistic research, but there is also intervention research. And one of, well, the most famous one is the finger study. It's a large study, and the intervention is a multi-domain lifestyle intervention. It's about diet. We just heard important things about diet. It's about cognitive training and cardiovascular monitoring, and it's about exercise. And within these interventions, and that was very interesting in the previous lecture, within these interventions, social interaction plays a strong but mostly implicit role. For instance, in this finger study, they do exercise, but and it's a group. So if someone is part of the group and is not joining several times, they contact that person and tell them, oh, we are missing you. We would like you to come back. Can we support you to come back? And things like that. And that really makes a difference. And because I asked the researchers, how is it possible that such a complicated intervention study, and we are so familiar with it, is successful? And they told me that this was one of the things that determined the success of the intervention, that they really were very keen on making the intervention as it really should be and including people in it, making them the owners of the intervention, that they learn that it's really important for them. And it turned out that this intervention was able to improve or maintain cognitive functioning in an at-risk group of older persons, which is also very hopeful. And this multi-domain finger study is now tested in a worldwide intervention. And coming near to conclusions, I will share this with you. I will simplify it. And what is also important, not only interdisciplinary research, but also looking at the neighbors, looking at research in longevity. And it turned out that for all many factors, including exercise, cardiovascular control, etc., they were all included in that study. But it turned out that the strongest predictor for longevity was to have a close confidant, someone you can share your problems, secrets, and pleasure, very important, and the number of chats during a day. And when I think about it, I get this picture that I'm presenting here. So in making your life more agreeable, you will have a higher chance of longevity and a lower chance of developing dementia. So in conclusion, social health markers have to take their place within research and intervention to improve the understanding of the dementia puzzle and to have an opportunity to develop related intervention. Thank you for your attention.
Thank you, Dr. Myra. It was a good presentation. Is anyone having any questions? Okay, I think they are not. So thank you very much once again. So we will move toward the next presentation, and it is from Laura Kim Finner from University of the Saarland, Germany. Hello, Dr. Laura.
Hello.
Am I audible to you?
Yes.
Yeah. So are you ready with your presentation?
Yes.
Yeah. I have make you the presenter. Please click on screen and share your screen and start your presentation.
You
Yeah,
can
it's
see.
done. Yeah, it is visible and you can start your presentation.
Okay. Moment. Moment. My screen is a little bit clogged by the information about the webinar.
Okay,
Moment.
it's no problem.
Okay. You see me?
Yeah, it's visible.
Okay. I can start now. Okay. Hello. My name is Laura Feiner, and I am a PhD student at the Saarland University. Our research is about aging and DNA methylation. Today, I will present to you some interesting results of our recent work. The title of my presentation is HOXA5 methylation changes correlate with age and gene expression. As most of you know, the aging process has been associated with differential patterns of DNA methylation, which also relate to changes in gene expression. Therefore, we aim to identify genes that are differently methylated between young and old individuals. We approached our aim by performing a genome-wide methylation analysis, followed by mRNA and protein expression profiling of the genes we aim to find. After all, our goal was to identify or find a correlation between DNA methylation and gene expression of a specific gene of interest. We started our approach by collecting blood samples of young and old individuals and isolating the monocytes from these samples. Next, we isolated the gDNA, mRNA, and protein from the monocytes. After a bisulfite treatment of the genomic DNA to convert unmethylated cytosins to uracils, we performed a genome-wide methylation microarray. This microarray analyzes the methylation status of over 850,000 known CpG sites. These are predominantly CpG sites, which are located in tumor-relevant regulatory regions, such as promoters or CpG islands. Based on this approach, we identified CpG sites which were significantly differently methylated in young and old individuals. We assigned the associated genes and selected interesting candidate genes based on criteria such as a methylation difference greater than 20%, a p-value under 5%, and the genomic location of the CpG site. For example, the promoter or gene body. Subsequently, we performed a targeted bisulfite sequencing of the CpG site. This is also called BiProf for bisulfite profiling of target regions. This step serves as a validation because here in the microarray, we have only about 10 to 20 reads per CpG site, and here we have over 1,000 reads per CpG site. Finally, we investigated the gene expression of the candidate genes on the messenger RNA and protein level by real-time PCR, ELISA, and Western blot analysis, respectively. The next slide depicts the genome-wide results of the microarray. Although overall methylation is known to decrease in age, the methylation at regulatory CpG sites, as analyzed by the microarray, tends to increase in age, as you can see on the left picture. The right picture illustrates the annotation of differently methylated CpGs, also called DMCG, in old compared to young individuals. In this picture, the left pie chart shows this distribution of the DMCG generated by the genome-wide microarray. As you can see in the two right pie charts, the DMCG, which are located in CPG islands, so in the blue part, are more often hypermethylated in old individuals than hypomethylated. And we have more DMCGs in non-island, in the orange part, in non-island regions that are hypomethylated in old individuals than hypermethylated. After performing the genome-wide analysis, we assigned the CPGs to the associated genes, and we chose 3 interesting candidate genes with a methylation difference greater than 20% and a p-value smaller than 5%. As illustrated here, the genome-wide methylation analysis indicated for all 3 DMCG a significant hypermethylation associated with old age. The 3 genes comprised dual specificity protein phosphatase 3, short DASP3. This is a protein that negatively regulates members of the MAP kinase superfamily, which are associated with cellular proliferation and differentiation, as well as homeobox5, short HOXA5, an anti-angiogenic member of HOX family, and the ryanodine receptor 1, short RUR1. This is a protein found primarily in skeletal muscle. We also selected them as candidate genes because the CPG sites are each located in different gene regions. The target CPG site of DASP3 is located in the 3' UTR. The target CPG site of HOXA5 is located in the promoter in a CPG island. And the CPG site of RUR1 is located in the gene body. After the validation with targeted bisulfite sequencing, only the target CPG site of HOXA5 showed a significant methylation difference, as you can see here. Next, we performed a real-time PCR on the 3 genes. It turned out that the methylation result was reflected in the mRNA expression analysis in the way that only HOXA5 mRNA expression is significantly downregulated in old individuals. This result is in line with the age-related hypermethylation of the promoter region of HOXA5. Next, we investigated the protein expression of HOXA5. As illustrated here, the HOXA5 protein expression is also significantly downregulated in old individuals, which is well in line with our HOXA5 mRNA expression result. As it is known that HOXA5 interacts with p53 and may upregulate the expression of p53, we also examined the expression of p53 in our samples. Since p53 protein is not expressed in monocytes, we only measured the p53 mRNA expression in the monocytes. As shown here, the p53 is also significantly downregulated in old volunteers. In addition, we see a middle strong to strong correlation between the HOXA5 and p53 mRNA expression. p53 is also known to interact with p10, an effective suppressor of cancer and contributor to longevity. Hence, we investigated the age-dependent p10 mRNA expression. In line with p53 downregulation, we observed a reduced p10 mRNA expression in old age. We also investigated the correlations of p10 mRNA expression with the mRNA expression of HOXA5 and p53. As you can see here, we have a middle strong to strong correlation. Finally, we studied the protein expression of P10, which was also significantly downregulated in old individuals and correlates with the HOXA5 protein expression. In summary, our results are as follows. First, we have shown that methylation of regulatory CpGs tends to increase with age, although the total methylation decreases with age. Second, our methylation analysis indicated an interesting candidate gene, HOXA5, whose methylation at the target CpG site is significantly increased with age, while its gene expression is significantly reduced in age. This reduction takes place on two levels, the mRNA expression level and the protein level. Moreover, we were able to show that the age-related regulation of HOXA5 gene expression correlates with gene expression of downstream targets p53 and p10. Our findings have shown how important it is to validate the results of a genome-wide methylation microarray by a targeted bisulfite sequencing, as we observed that after the validation, our number of putative candidate genes become less. Our main result, that age-related hypermethylation of HOXA5 correlates with its reduced gene expression, qualifies HOXA5 as an eligible aging marker. We further conclude that HOXA5 hypermethylation in advanced age contributes to p53 downregulation and, in that context, may also contribute to the increasing age-dependent cancer risk in the elderly. Thank you very much. Do you have any questions?
Thank you, Dr. Laura. It was a nice presentation. Is anyone having any questions? Okay, I think there are not. So thank you once again. So we will move toward the next presentation. It is from Dr. Eof Hogerwurst from Loughborough University, United Kingdom. Hello, Dr. Eof.
Hi, I'm trying to.
Yeah, I'm going to make you the presenter.
Thank you.
Yeah, it's done. Now you can share your screen and start your presentation.
Start broadcast.
Yeah, it's visible, doctor.
Okay. Thank you so much for inviting me. Can you see my talk as well?
No, the presentation is not visible. It is showing that some screen broadcast GoToMeeting.
Okay.
And it's loading.
And I just sent you my talk, didn't I? Because if you could open.
Can I know the mail ID to which you send the presentation?
That's. Oh, wait a second. I think it's now sharing my screen.
It's in process, right?
Mm-hmm.
Yeah, it's visible, doctor.
Yeah, can you see this?
Yes. Yeah, it's visible.
Let me do it like this. It's okay.
Okay.
Thank you so much. Thank you very much for inviting me to speak about lifestyle and dementia risk. I work at Loughborough University and at several universities in Indonesia. And I'd like to disclose a review I did for Procter & Gamble on B vitamins in 2019, which I will be presenting here. Loughborough University in the UK is in the top 5 of UK university and is #1 in the world for its sports and exercise research. As you can see, we are in the heart of England, about an hour from London. And we investigate dementia risk and protective factors worldwide. So we have cohorts in Indonesia, in China, various cohorts in the UK and Europe we work with, and in the US. There is no cure for dementia, so we can only focus on prevention of dementia. And that is in 3 levels. So primary, over the lifespan, to prevent the buildup of pathology. Secondary, which is once people have developed mild cognitive impairment, which is a risk factor for dementia. And tertiary, once people have dementia, to reduce the worsening of symptoms. So there are things we can do to change our dementia risk. And my take-home message is, what is good for your heart is good for your brain. So these are the risk factors for dementia: an older age, being of the female gender, in most cohorts, not all, having little education, being poor, genetics, and then there are things we can change, such as not smoking, having a good diet and exercise, which I will be talking about in the next couple of slides. And these factors can prevent, in midlife, being obese, having high blood pressure, high total cholesterol, and being diabetic, which are known risk factors for heart disease, but we also now know that they increase the risk for dementia. This might explain the difference in gender between cohorts, because men tend to die earlier of heart disease, and so they cannot then go on to develop dementia because they share the same risk factors. As I said, poverty and economic deprivation is often a separate risk factor and is associated with smoking, excess alcohol intake, having a poor diet, and low levels of exercise. A meta-analysis, a recent meta-analysis, showed that these lifestyle factors can really increase the risk for heart disease. But particularly, and this was so interesting, so this is the overview of that meta-analysis. What it showed was that these risk factors play a very important role in midlife, but not so much in later life, to double the risk for dementia. And sometimes more than double the risk for dementia if you carry more than one of these risk factors. Now, what's interesting here, and so we need to look at primary prevention when we look at risk factors associated with heart disease. What we see is 15 years before the onset of dementia, we see that high blood pressure is a risk factor for later life onset of dementia. But then data show a decrease in that blood pressure about 1 to 2 years before the onset of dementia. And the same seems to happen for cholesterol. So in midlife, 15 years before the onset of dementia, high total cholesterol is a risk factor for dementia, including Alzheimer's disease in later life. But then a couple of years before the onset of dementia, cholesterol starts to decrease. And also body mass index. So we often see midlife overweight being a risk factor for dementia. But then a couple of years before the onset of dementia, the obesity starts to decrease. And often we actually, like in Oxford, we found lower weight in people with dementia. So what's causing these risk factors in midlife? Well, several data showed that high blood pressure, for instance, in midlife, was associated with childhood IQ. And below is a model that might explain this. So we have childhood cognition leading to, which is associated with optimal environments, having access to resources, including better foods, more socially stimulating activities, music lessons, additional courses, etc., building up cognitive reserve, which creates an optimal environment. optimal educational attainment, leading to better socioeconomic status and less risk of the metabolic syndrome. So this is hypertension in midlife, abdominal obesity, high triglycerides, low HDL cholesterol, and an increased risk for diabetes. So this is in midlife. And what we found was that, because that would sort of make you think, well, what's the point of prevention if it's so determined by high or low socioeconomic status? And indeed, a lot of studies find that people who are rich, who are affluent, the middle classes, who are of high socioeconomic status, have better diets, and they have lower body mass index, they exercise more, less risk for cardiovascular disease, more access to medication, they have holidays abroad, exposing them to better D vitamins, etc. But in a recent review I found that people had less cognitive decline if they were poor, but when they ate good diets. So this ties in with the talk that we saw earlier on fruit and vegetables, and not eating so much fat, certainly not red meat, not lots of high fat. Those people actually had less cognitive decline. So despite being poor, having a good diet can offset that risk of poverty across the lifespan. Now, if we look at these macronutrients, we find, for instance, that cholesterol intake, but also statin use to combat this high level of cholesterol, can reduce the risk for dementia, but there are several studies that do not find this. And one of the problems is that people usually start taking statins and make that part of other lifestyle changes, so they also start exercising more. Other fats, good fats, such as omega-3 fatty acids, are quite controversial in terms of how much they affect the risk of dementia. Now, 10 out of 14 studies found that taking omega-3 fatty acids benefited people who are at risk for mild cognitive for dementia, who had mild cognitive impairment. So this is secondary prevention. And the best seems to be to have this intake through fatty fish, such as mackerel and salmon and sardines, and eating nuts. Another good fat can be found in olive oil, because olive oil contains oleocanthal, which can fight the plaques that we see in Alzheimer's disease. But olive oil can also improve the immune system, which is known to play a role in dementia, improve vascular health, which, as I told you, is a risk factor for dementia, and reduce the risk of dementia altogether. Now, there was a study in over 500 elderly at risk for cardiovascular disease who were given a Mediterranean diet with lots of olive oil and nuts. And what they found was that that reduced the risk for cognitive impairment and dementia. Now, if we combine olive oil with a green salad, we are creating nitrosamines, which can further reduce blood pressure. So this Mediterranean diet seems to play quite an important potential role in the prevention of dementia. And we did some reviews showing that this was indeed the case. People who ate the Mediterranean diet had lower rates of cognitive decline and a reduced risk of dementia. And also a recent meta-analysis of 7 randomized controlled trials and 38 longitudinal studies showed a reduced risk of global cognitive decline in middle-aged people and older people, but not once they had developed dementia. So this is important for tertiary prevention. Once you have dementia, it's too late. So what are we talking about? Should we all eat more tiramisu, lasagna, spaghetti bolognese, quiche? No. This is about the traditional Mediterranean diet with very little animal protein, very little sweets, but lots of olive oil, vegetables and fruits, and whole grain carbohydrate. These whole grain fruits, grains and fruits and veg contain antioxidant vitamins. Now, again, taking these vitamins as supplements is quite controversial, whether that really protects against cognitive decline and dementia. And one of the reasons might be that a lot of these food supplements often contain iron and copper, which probably isn't very good because that could play a role in oxidative stress. There's been some evidence of vitamin E being protective, but again, this is best probably in whole foods such as nuts and seeds, whole grains, and green leafy vegetables. Now, these whole grains and green leafy vegetables found in the traditional Mediterranean diet also contain lots of B vitamins like folate. And this can reduce homocysteine, which is a risk factor in dementia. These are data from Oxford showing how low serum folate is found in people with Alzheimer's disease compared to controls. But studies that try to, again, give supplements have not always found benefits of B vitamins in reducing cognitive decline. Now, there might be a case that cognitive decline can only be prevented in people who were given folate who were actually folate deficient or at cognitive impairment. And these are 3 studies showing that if you give these B vitamins to people who were actually folate deficient, that there is a beneficial effect on improving cognitive performance and in reducing hippocampal loss, which is associated with memory loss and an increased risk of dementia. I did a meta-analysis, this is the meta-analysis I did for Procter & Gamble, to look at secondary prevention. It seemed to be from our earlier work that once people have dementia, again, giving these supplements did not prevent worse dementia. However, in people who had mild cognitive impairment and who were folate deficient, 4 out of 7 studies showed beneficial effects. However, as you can see, this beneficial effect is very small. It was significant, but it was tiny. So while diet may have preventative effects, exercise is probably most important because this can affect cognitive function over the lifespan, including once people have developed dementia. Why? Because exercise can have beneficial effects on these cardiovascular disease risk factors, but also stimulating blood flow to the brain, regulating blood glucose, reducing the risk for diabetes, which is a risk factor for dementia, and it can promote dendritic outgrowth of the neurons, also in older animals. And this is very early work by Diamond et al., showing that if rats were exposed to stimulating aspects in their cages, they started running around trying to explore this, and that promoted dendritic outgrowth, also in these older rats. We did some work in Indonesia showing indeed that older people who engaged in sport had better cognitive function on those cognitive functions that are sensitive to dementia, and that engaging in sport halved dementia risk. And these are data from our study that we're involved with called the PRIDE study, where we showed that also before people had developed dementia in midlife, when they engaged in lots of physical activity, they had a lower risk of dementia between 30 and 50%. And that the more physical activity they engaged with, the more reduced that risk was in a follow-up over 10 years. But also once these people had developed dementia, keeping physically active reduced the rate of decline. And this is a study in tertiary prevention, so once people had dementia. And what we can see, it's a Dutch study, was that over a year, these people who were asked to do stretching and strength exercises had a much improved global cognitive performance compared to controls who went down. We did a review in 2017 also showing that the majority of studies found beneficial effects on physical activity on cognitive function, even once people have developed dementia. Now, there have been a couple of studies recently showing that if older people with dementia were exposed to very strenuous exercise, there was no benefit of global cognition on global cognitive function. Now, these people in some of the studies, like a study by Sarah Lamb, she actually found slightly worse cognition. And the question was whether the exercise was too much. We've consistently found in our strength exercises, which can be found on our website under Couch Potatoes for Cognition, that if you are engaging in strength exercise, you can have improved memory function, whether you have dementia or not. However, more research needs to be done. So while this meta-analysis did indeed find that physical activity, including resistance training or mixed physical activity with aerobic components, did improve global cognitive function, there was lots of methodological issues between studies, and so more studies need to be done to show how much we need to do and of which intensity. So in conclusion, the risk factors for Alzheimer's disease are the same as those for heart disease, such as high blood pressure, smoking, having diabetes, high total cholesterol, being obese, lacking folate and vitamin B12, and these risk factors need to be treated in midlife. Because once you develop dementia, these risk factors tend to decline, or as is in the case of folic acid, you can't really use these treatments for tertiary prevention. The best is probably to take a Mediterranean diet, which has some benefits, such as we heard earlier, in Thailand, containing lots of fruits and veg and whole grain carbohydrates. There are possible beneficial effects of exercise, combined exercises, mixed exercises, including strength exercises for MCI, so the people who are at risk for dementia, for secondary prevention, but also in tertiary dementia, so once people have developed dementia, on global cognitions. And one of the other benefits we see is that during exercise, we found this in a study in Portugal, that people have a reduced risk for falls. Now, falls are, the risk for falls is doubled in dementia, and each fall will cause worse cognition in dementia. So it's very important to treat this and prevent this, because the risk for falls leads to hospitalization, that often leads to delirium and other psychiatric symptoms, leading to more medication, leading to increased care burden, etc. So decreasing falls is super important. One of the risk factors for falls is changed gait. So people have a slightly changed gait in dementia. But also what we see is that there is a loss of muscle mass. And so by providing exercise with adequate nutrition, we can reduce that risk for falls. I'd like to acknowledge my collaborators. So this is my staff in Indonesia, which is led by Professor Tribhuti Rajaharjo. And I'd like to also thank my collaborators in Oxford, Professor David Smith, and in Cambridge, Professor Brain. The people I worked with in UCL have now moved to Nottingham, Professor Orrell and Professor Blossom Stephan. And of course, the people who supported us with grants from the NIHR, the MRC, the Wellcome Trust, and various others. And you for listening. Thank you so much. Do you have any questions?
Thank you, Dr. Hauser. First, it was a nice presentation. Is anyone having any questions? Okay, thank you once again, Doctor. It was a nice presentation.
Thank you so much.
I think there are no questions from anyone. So we can move forward for the next presentation. It is from Dr. Marcelo Menapace from M&M's Consulting Limited, UK. Hello, Dr. Marcelo. Are you ready to give the presentation?
Hello. Hi. Yes, I'm ready. Can you hear me?
Yeah, it's audible, Doctor.
Okay.
So I'm going to make you the presenter. You can share your screen and start your presentation. Yeah, it's done, Doctor.
Okay, let me start it. Let me know when you can see my screen.
Yeah,
Perfect. Okay. Thank you very much for your time, for the invitation today. I would like just to talk about nutrition in general and how and why we are what we will be called in the future holobiont. So the summary is just a cool way to take out the message, which is generally what is a holobiont and how come we are holobionts. So we need to discuss the nutritional aspects of these, which are generally the prebiotics. And then I'll come into the discussion with blood types and how these are all together related. And finally, some examples to go to the conclusions and some dietary tips that we can all use. So what is a holobiont? In general, a holobiont is a mixture of different organisms living inside one single organism or species. In actuality, we can say that it is what it can be called a meta-organism, meaning that microbes or any kind of viruses or any kind of other microbial organism can live inside of some niche inside of the body of a bigger species of what we will call that the actual general species or the person that we want to identify. And that being said, it is very important to underline how many different species are present in any single organism. So if we consider an animal, whatever animal we want to, let's say, for example, a bull, okay, that species will have inside itself trillions and trillions of different organisms. Can you still hear me? Okay. So in general, when we consider a holobiont, we consider all the species together, and they grant the holobiont, that organism, with other different enzymatic capacities above and beyond, greatly above and beyond what that single organism would have without the advent of the microorganism. So we can enumerate as many as we want, and these can finally produce what is the end result of any kind of product that we desire, that our body or the body of the organism of that single creature would desire or would need. So let's make an example of exactly how much are these microorganisms, just to be clear. We thought previously that the human body only had microorganisms, at least 40, 50 years ago, only in the intestines. So that's where we initially thought that all the external organisms present in our body would be. So in a small niche, these are the places where they are. But then we started to look around, and we found out that it's not only in the large intestines, already around every other internal organ. So it's the intestines, it's the stomach, it's the mouth and the throat. So all our digestive tube is filled with these microorganisms, each with their special numbers and diversity of species. Of course, the types of microbes that are present in the intestine are not the same that are present in the stomach and not the same that are present in the mouth. Generally should not be like that. But then we went on to search other parts of our body, and well, incredibly so, we found that even the respiratory system has its own microbiome, which is a special suit of microorganisms that live together with us, and we provide them food and nourishment, and they provide us other important things that they can do. For example, other enzymatic capacities that they give us. We find them on the skin. These are normal. They have to be there. Without them, we could not survive. We find them in our blood, incredibly so. We thought that our blood would be completely sterile. That is not so. There are microorganisms, not just viruses present inside the cells, but microorganisms present inside the cells and even present outside that have to be there to function, for us to function perfectly. We find them also in the brain, incredibly. This has been demonstrated now for over 20 years, but yet the information has not really went out, because if it were to say to go out, and this would change really the way medicine looks at every single human body in this case. Another important thing is what are the micronutrients or the nutrients that these microorganisms use. We call them generally prebiotics because they come just before the life. They normally use, so all the microorganisms normally use to consume these probiotics. And these prebiotics are generally glycans. Glycans are special types of carbohydrates. These are carbohydrates, of course, they are all carbohydrates, but special carbohydrates in the sense that they are able, with their enzymes, to destroy or to break the structure of these carbohydrates. Now, special carbohydrates, because they have with a difference with respect to our normal carbohydrate that you consider, which is glucose with alpha kind of bondage to another glucose or to a fructose. So these are the normal kind of carbohydrate that we consider, sucrose, other carbohydrates like that. But there are special kinds of carbohydrates that have to be considered, and these are generally called as glycans. And the glycans are what are normally used by the beneficial bacteria or the other beneficial kind of microorganisms present in our body. As we can see, they are fundamentally used by these microorganisms and are transformed into further carbohydrates or other substances that are then capable to being utilized by the body itself. These are some illustrations of important glycans that are present in food that we eat, and we may not know about it, and are present also in our body. So we produce them, and because these are present generally on the membrane, the cell membranes of all of our cells. This is just a legend to understand what are the standard terminology and the standard structures of all these glycans. Now, differently with respect to proteins and, of course, to DNA and RNA, the normal disoxyribonucleic carboxylic acids, because of their capacity to bind in several different points each and every single monocarbohydrate, so monosaccharide, they are able to branch out, and this means that they are extremely more complex than proteins. While proteins are just linear, these can become extremely more complicated. Now, these are very simple ones, okay, just one branching out of them, and we have beta kind of bondage, means that they are more complex and they're sturdier to digest. For example, the first one is a clear example of cellulose. We are not able to digest cellulose, but certain bacteria are capable of doing that because they have special cases, special types of enzymes that are capable of destroying this kind of bondage. Both the B14, also called beta-1,4, or the beta-1,3, or beta-1,2, or beta-1,5. Other carbohydrates have different structures based on the different monosaccharides present in it. These are some examples, and these are more complicated examples of these glycans. Now, of course, if we cannot digest them, then they have been termed fibers. So these are present whether in algae, whether in other fruits, whether in other foods that we eat, it doesn't matter. In general, because we are not able to destroy them, then we are not able to absorb them. They are defined as fibers, and of course, they have the function of fibers. And this is another example, a very simple kind of change sometimes, but most of the times are much more complicated and much more difficult to get at or to destroy, even for the enzymes present in the microorganisms. Now, why are we considering then all these glycans that are present in our body, and why are not we putting this together with the fact that these glycans are able to be recognized by certain kinds of proteins present also in our body, present also in the food, but also present in and on the cell membranes or the cell barriers of the microorganisms. And these are called lectins. So lectins are special kinds of proteins that are capable of recognizing those specific glycans. Now, most important among all of the glycans present in our body are the ABO grouping glycans. Because of their structure, they are present. We thought that they were present only in the blood, or only on the red blood cells. But in actuality, we find them all over our body, in every single cell of our body, not just on the proteins there, but also on the also on the lipids. So we got also glycolipids that have the terminal part of one of these four groupings. Now, we all know that the human body can be defined as being either one of the four groupings of the ABO blood type system, because of its importance is one of the highest among all. Other groupings of blood typing is based on other proteins, but these proteins are not present if not on red blood cells and do not have any other function like that. But the glycans, these special glycans, the ABO glycans, not just present on the proteins, not just present on the lipids, but they're present everywhere in our body. And we can also express them outside of the cells themselves. For example, mucilins are special proteins that are emitted and secreted by our intestines and or the whole digestive tube. So they can be attacked by lectins outside of the cell and in a very close contact with the tissues that we have in our body. Among these, we all know that the blood type O is the less complex, and we will now see one of their structures, but the most complex are definitely the A and the B. Of course, there are people that have both of them, A and B, and they can have different reactions, very similar to both an A and a B. This is an example of a cell membrane where on top of the lipids and on top of the proteins, we have glycans that are present. These are ABO glycans, but can be also other type of glycans. In general, what they do is to react better or to help proteins and lipids react better with other proteins, glycans or lipids, whatever that specific protein lipid has a function. So it doesn't just stabilize or render more stable the protein, but it also gives to the protein special functions. So these are other examples of glycans. This is the fundamental structure of the ABO blood groupings. So at the end of every glycan, it may be of just 3 or 4 monosaccharides, or it can be a very long chain. It doesn't matter. But at the end, we have the blood type A, the blood type B, and we also have the blood type O. Every single body has one or at the maximum 2. So a blood type, an AB blood type will have both the first and the second. But in general, would have one of these. And based on this, of course, the reactivity of the structure of this chemical compound would differ and would give and provide a different reactivity to the protein. That's why people who have different blood types can have some susceptibility to diseases that are different. Now, one example of this, which was known in the past, especially in the 1960s and 1970s, is the fact that blood type O have a higher acidity of the stomach with respect to other blood types. And this has been demonstrated for over 60 years now, since the 1960s, we're in the 2020s. But it is very difficult for a medicine person, an expert in medicine or a physician or any kind of even nutritionist to understand the complexity of these differences. We in general have been taught, as you can see, that humans have an acidity, an overall acidity, and an empty stomach of around 4 pH 3 to 4. In reality, what we need to see is that when we do experiments, and this has been done from the 1960s, even before that, we see that the actual pH of the stomach is around 1.5, where it is there especially for the blood type O's, while the other blood types have a slightly higher pH, but it goes around 2.5. So this brings us to the understanding that we may fall into the overall trophic grouping of the carnivores. Now here we have an example of the blood type O's and the difference with respect to the blood type, the ABs or the other blood types. And you can see the scale is generally the basal acid production in milli equivalent on per hour. So every hour we generally manufacture and throw into the stomach that amount, which is good, of course, when we have just eaten. So we have eaten proteins, okay, that's good. We throw in any amount of acid, and slowly we start to digest those proteins. When it's done, it goes ahead back into the further intestine, and then it goes on. But the problem is that when we do not give them the right proteins, because the acid present in our stomach combines with the pepsin that is secreted in our stomach in order for that to be activated. And once it's activated, it can then destroy the proteins. If we do not have enough proteins there in our stomach, then this amount is not reduced in terms of production. The same amount will be produced, but then it gets wasted and it has to attack something, and eventually it'll start to attack our stomach, our intestines, or whatever else. And then the end result is the production of ulcers or other diseases of the gastrointestinal system. So in practice, we need to take into consideration the fact that there have been a lot of studies around the interaction between microbes and the blood typing present on the cells of our body. So the glycans there can interact with these microbes. And the end result of this is that there are certain microbes that are better suited for blood type O, other that are better suited with other blood types. Therefore, we have a diversity of also in microorganisms. This has been studied for at least 40 years, and we should be able to put this into consideration. Another fact is that we are not just a blood type A is just a blood type A. That's not correct, because there are several different subtyping there. So this increases the variability and increases also the diversity with respect to what a single person or another person can or cannot do in terms of reactivity. One final point before we conclude is the fact that independently to the blood type A, B, or 0 or AB, we all need a good amount of vitamins and minerals in order for our companions, microorganisms, to do their job and for us to do our job. So we have been told that the recommended daily amount is the right amount that should be given. Now, it doesn't matter how they have come up with this. One thing that we should consider is the fact that often we have we know there are limits above which, if we take that specific micronutrient, we will start to have an adverse event. But that being said, we can see here that there are several vitamins that do not have this upper limit, has not been studied, or it has been studied but has not been found. This means that between the RDA and the upper limit, whether it exists or it does not exist, we have this space of, let's call it therapeutic option or therapeutic help, where we can use the micronutrient at our disposal in order for us to absorb it better, first, and second, in order to replenish all the reserves that have been depleted after years and years of eating extremely depleted foods, as we all know. In the end, I would say that since we are more than 90% of our body in terms of weight, when we take out water and we take out the minerals, over 90% is our proteins. So we should focus on proteins. And another reason is because we are able to create carbohydrates or create energy out of proteins, out of fat, but we are not able to create proteins out of carbohydrates. So that is a limiting factor. We don't need carbohydrates because physically our reserves of carbohydrates are 2%, 3% at best. What we transform them into, generally fat. So we should be concentrating and focusing more on proteins, which are the essential elements, together with the other micronutrients, vitamins and minerals that are needed. And then if we can consider sometimes, if we are especially overweight, fasting, that does certainly help out also our immune system, especially in terms of the growth of the bacteria present in our intestines. And thank you very much for your time today.
Thank you, Dr. Marcelo. It was a nice presentation. Is anyone having any questions? Okay, thank you once again. I think there are none. So from this, now we are ending the session before the lunch. Now we can take for the lunch break, and we'll be back at 13:40.
Thank you.
Thank you very much. Yeah, hello, Dr. Rolls. I'm making the
Hello.
presenter.
Yes, so now I want to see the note only for myself and to show the slide for everybody. So
Okay. No,
how
whatever will
can
be
we
in
do
your
it?
slide, it all will be visible to us.
No, there is an option in the Zoom. I know that there is option that I will see the note and the slide, but the audience will see only the slide.
Okay, I think there is no option like that in go to meetings. The only thing when you
If
share the screen, yeah, this
if I
thing.
do like this, you see all
Yeah, I'm screen.
options,
The
make
notes are
present,
visible to
include
us.
media sound, remote, control draw. No, there is something. No, we have to
Here
do.
in this, all will be visible to us.
No, so there is an option. You want to know how to do it?
No, there is no option like this in GoToMeeting that half of your screen is visible and half of yours is not. The total screen of yours will be visible to us.
You sure? Erase, check, attention, normal, remote, options, include media sound, make presenter. So now you see everything.
Yeah, everything is visible.
About go to support. So you see like this. Okay. Okay. So like this, okay?
Yeah.
Mm-hmm. Okay. You record everything.
Yeah, everything will be recorded, and once the webinar is over, we will send you the recording and it will be uploaded in YouTube. So you can access your webinar like video presentation from YouTube also.
So you send me separate my or you send everything
No, it will be our total.
total.
Yeah.
Okay. Okay. So it will be
Okay.
like this. Now you can see it
Yeah, it is visible.
and you see everything.
Yeah, everything is visible.
Okay.
Okay. So in a few minutes, I will be making the presenter. I will be calling you and will make you the presenter. You can start your presentation once everyone is back with after the break.
Okay. Thank you.
Like there's 2 to 3 minutes more. That's
Okay.
it. Okay. Thank you, doctor.
Thank you very much.
Yeah, hello everyone. Welcome back to the webinar. So we can proceed towards our presentation after the lunch. It is from Dr. Ozur from the Israeli Center for Dizziness and Balance Disorder, Israel. Hello, Dr. Oz.
Your voice is not, your voice is off. Dr. Oz, your voice is off.
Yeah, Dr. Oz, it's audible.
Like this, now it's okay?
Yeah, it's clear. And you can
Okay,
start your presentation.
good afternoon, everybody. Thank you for the organizer for inviting me to speak about how can we prevent the next fall. I'm a physical therapist, almost 30 years. Developed the teaching to balance approach for imbalance, for improve balance and challenging the sensory integration with respect to psychosomatic influence. I see a patient in the clinic in Israel, and I also teach in the master degree in University of Ben Gurion, a student, a physical therapist for vestibular rehabilitation, and a founder of the Be Balanced application, assessing balance and training in goal to improve balance and decrease falls. We know that people aged 65 and older represent 10% of total population in Israel, 14 in United States, and 18% in Europe. The number of people 65 and up is increasing and is expected to be double in the next 20 years. Instability is one of the three main issues older people encounter every single day. Here are a few statistics about dizziness, imbalance, and falls in the elderly. About 65% of people above the age of 65 have a decrease in the sense of balance. Every third person over 65 falls once a year. Approximately 15% of falls end with a significantly traumatic injury that needs medical intervention. And we know that fall lead older people to emergency room, disability, low self-esteem, and death. All this information is all over the world. Falls are related to extrinsic and intrinsic factors. The extrinsic starts with slippery carpets, through obstacles like cables or shoes on the floor, the evening twilight, when shadow confused one's vision. The intrinsic factors are related to illnesses, dysfunction, and recalibration after absorbing sensation from the three sensory systems. And we know these three sensory systems is the visual system, the vestibular, and the somatosensory system. All of them function together to keep our postural control in alignment. In 2006, Rubinstein and Josephson demonstrate the multifactorial cause of falls in the elderly. They divided them into intrinsic, extrinsic, and precipitating causes. Vestibular dysfunction and balance, as you see here, balance impaired, are cited in the first place. And I claim that they are the most important cause of falls in the older people. At the same year, we published our understanding after examining 169 individual independent elderly living in a community and in retirement homes. 84 out of them were operated due to hip fracture after a fall. We diagnosed them in less than 2 weeks after the hip operation. Most falls were related to dizziness, vertigo, and unexplained imbalance. The control group was 85 elderly people who were living in 4 retirement homes with no reported falls in the last 6 months. And the conclusion from this study was that the vestibulo-ocular reflex, the VOR, was significantly decreased in the group who fell and broke their hips. Why? Was it deficit in the VOR before or after the fall? Is the VOR deficit the main cause of hip fractures? We believe that VOR is an important player to keep our postural sway or postural control. And even when there is a fall already happening, the functioning of the VOR may lead to accurate response, which might be the difference between a minor or major injury after fall between one to another. Furthermore, we believe that VOR deficit might be indicated as a marker of high-level disorder in older population. Kale in 2021 reviewed 13 studies with the goal of finding 2 studies that emphasize the same risk factor for a fall. The list you can see here relate to these positive factors. When they mention balance problem, it is a little complicated because there are plenty of clinical tests for assessing balance. As a physical therapist, we know that there are many different clinical tests. So before looking at a different test for assessing balance, let's go and see the main system responsible for balance, which is the inner ear. So the ear, we can see here the outer part of the ear until the eardrum, the middle ear inside with 3 small bones, which translate sound wave to be absorbed in the cochlear area, and the inner ear here in the small rectangle, the size of which is approximately the size of neutral nail of your little finger. And if we enlarge the inner ear, you can see the 3 semicircular canals full of fluid that gives us information about angular head movement, acceleration and deceleration. The 2 compartments here, the sacula and the endoauricular, which are full of crystals, gives us information about linear head movement. With each direction of the head movement, the vestibular system is an important partner to respond very quickly to help to keep our balance stable. For example, if we turn the head to the right, the fluid inside the canal will move to the left. And the small hair cells that you see here will move to the left as well, and it will start the reflex arc of the VOR, the vestibulo-ocular reflex. The eye will move to the other direction if the system works good. But as I started, 67% of the elderly might have the deficit in this system. Why all of this is very important? So there are many clinical tests to assess the balance. I would like to see which of the tests will examine also the vestibular system. So most of the test are not sensitive enough to capture mild instability. The main reason is absence of vestibular assessment. It means that elderly come to the clinic, say, I have no balance, and when I do some of these tests, I will not find the imbalance. I say, no, you are very good, you get 95%. Why it's like that? Because the reason there is no assessment for the vestibular system. These tests are better for people when they have low balance function, and for population with other comorbidity, like, as we heard in the morning, the Alzheimer, maybe CVA, Parkinson's disease, MS, or some other diseases. When we want to assess the healthy elderly population with mild to moderate balance impairment, we have to consider the vestibular system as well. So if we look, these 4 tests, the time up and go, the Berg balance scale, 4 square, and single leg balance, these tests do not include head movement. So if they don't have the head movement, they will not examine the vestibular system. Maybe the static, let's see. These 3 clinical tests, the modified clinical test, standing on a form, eyes open and eyes closed, and on the floor, eyes open and eyes closed, dynamic gait index, walking 20 foot on the hall, stairs up and down, bending, walking and turn the head, and the Zur balance scale, which is standing in different position with head turning left and right, up and down, eyes open and eyes closed. All of these are checking, assessing the vestibular system, but not all of them do it dynamically. So if we look for dynamic head movement, we need head movement to do as well the test. So the first one is also not included. Only the DGI, the dynamic gait index, and the Zur balance. But if we want only a small space, a balance test for standing position, and I don't have a long hall to walk with the patient, and I don't have stairs, I just want to stand on one place to measure the balance and to know the risk for falling. It's the ZBS, Zur balance scale. So if we look what it's about, we in 2017, we demonstrate our understanding after 2 years of follow-up of 75 healthy older people. All of them were administered an oculomotor clinical examination, the ZBS, respect to predicted cutoff, and also we did also the Berg balance scale with the cutoff of the balance scale, the number of comorbidity and medication, and the conclusion of the study was that the number of prescribed medication, history of falls, and the ZBS score were the predictors for traumatic falls. After 2 years follow-up, and we said that if you got a cutoff under 51% of the ZBS, you will fall and maybe will broke a hips or nose or something else. And after follow-up 2 years, what happened with those population? It was submitted and advertised. So an innovation telemedicine. The last 2 years, we have a company that take all the idea and try to make an application, a vestibular rehabilitation training at home, anywhere, anytime. specifically designed for older people with aimed of improving balance, preventing the next fall. And the B-Balance application save money because you can start immediately. Lifesaver because you go out from depression and anxiety and fear of falling and reduce movement to do the exercise that will be accurate, tailored for your situation. Accessibly, it's very low cost. It's for everybody from everywhere for any time. Application self-assessment and training. You have to be present 10 exercise to evaluate your balance and then take off your shoes, and it's written that press here to continue. And if your balance is in the middle between mild to moderate, you can do it alone. If you have a very low balance ability, you have to go back to your physiotherapist and do it with him. Pilot study, I would like to finish with this. A pilot study of 41 independent elderly used the application. 28 were using the B-Balance application with the blue bars as you see here. And 13 with the red bars didn't do the application, just do the examination at the beginning, the ZBS. And what we can see from preliminary to the baseline, we have no intervention. And there is no difference in both groups. But from baseline time to the end of the study, only one month, twice a day, using for 5 minutes the application, doing the exercise, the B-Balance group improved from 54 to 63%, which means that they shift from high risk for falling to moderate risk for falling, compared to the control group that stay at the same 4 months. Twice a day, 5 times a day, tailored exercise make the difference. Thank you very much and enjoy from the webinar. Thank you for all.
Yeah, thank you, Dr. Rose, for your presentation. Is anyone having any questions? Okay, thank you once again. I think there are no questions, so we can move toward the next presentation. It is from Martin van Leen from Martin Geriatric and Wound Consultancy, the Netherlands. Hello, Dr. Martin.
Hello. Can
Hello.
you hear me?
Yeah, it's audible, doctor. So are you ready
Okay.
to give your presentation?
Yes, I need to
Yeah, I'm going
start
to
my.
make you presenter, so you
Yeah.
can share your screen and start your presentation.
Okay.
Yeah, I'm making you the presenter. Yeah, your screen is visible now. Yeah, now you can start your presentation.
Let's hope. Okay. Thank you very much. I'm Martin van Leen. I'm an elderly care physician in the Netherlands, and I'm very interested in wound healing. And one of my topics is that I did a study about Alhydran moisture cream for health skin problems. Why did I do this literature study? Because I think it's very important that you use the right products. Normally for skin problems we use corticosteroids, and they have a lot of problems by using them for a long time. And still there is a lack of consensus regarding the most optimal treatment of a disrupted skin barrier. As we look at the skin has two main functional layers, the epidermis and dermis, which leads to protection, terminal regulation, absorption, and other endocrine productions. This is a picture of the skin. The top layer where I'm now are dead cells. There is a small layer in between, a very important layer, because there are a lot of proteins are made there, which make the best result for water control inside the skin. And below we see that there is a lot of cells who are coming from the bottom until the top, and there are also some cells who have a very important role in the immune system. The skin barrier has four important functions. The basal barrier, done by the stratum corneum and epidermis, the biochemical barrier, which protects the pH gradient and the calcium gradient, the immune system, and a barrier who is able to get a good hydration of the skin. And I'm only speaking about point 1 and 4 because that are the functions who are important for a good water balance inside the skin. As you see, as I already told you, the upper layer, the stratum corneum, is the main barrier. And below, there is a very important function called transepidermal water loss system, which results in a good hydration of the skin. Transepidermal water loss is also controlled by the natural moisturizing factor, and factor is one of these humectants, proteins that attract and bind water from the atmosphere to the inside the skin, and also regulate some getting water from below. Why is this important? Because a proper hydration of the skin may result in a better plasticity of the skin, protects it for damage, and controls the function of the corneum, and altogether results in a skin barrier which, when there is a lot of loss, results in severe itching, redness of the skin, dry skin, and also skin defects. But not only skin defects, also the healing process will be not in a proper way. In this slide, you see what's happening in normally. Normally, there is coming water from below, out of the vessels in the dermis, and this goes to upside, and then at the top it will get out of the skin mostly in a sweating form. From above, the skin can be damaged by UV rays. That's why we need a lot of protection when we are outside in the sun. But also it can be damaged by irritants, and a lot of people use much too far irritants because they are all when you are going into the shower. When the balance is not all right, then you see that the upper layer of the skin will be damaged.
Çocuğundan geldi. Çalıştı, geldi. Ama benim bu kafama göre bir şey olmadı. Şimdi
I hear some translation. Is it correct? Okay. This is a picture of a damaged skin. this is mostly related to age,
9 istasyon olduğu
but
için
dry
geldi
skin
mi?
diseases
Gördün mü?
like psoriasis and contact dermatitis or hypostatic dermatitis are all other
9 istasyonda
ways of
çalıştığımız için
looking for
tabii istasyon verisi çok az. Ben aslında
Wait a minute, Dr. Niloufer has to close the microphone.
hem de air quality rapor
Thank you.
Okay. What are the problems with skin diseases? They may result in extensive water loss. The lipid production inside the skin will be bad. It will get to a drier skin, and what's very important, a higher pH at the top of the skin. And that may together may result in higher risk of infections. But not only what I said, also when you look at a scar, delayed wound healing will be there when there is more loss of water than wanted. Mr. Musto found out that the balance should be all right, or else there should be an increased collagen production, what we often see when there is scar formation or even when there's keloid formation. So it's very important to restore a disrupted skin barrier. And for all the three diseases as shown on this slide. What is a need for a good treatment? And I'm sorry, there is a type of failure from me. A good treatment should have occlusive agents and on top, so that it's impossible that irritant from outside can damage the skin. It should be having a good water from the deepest layer, and the emollients should be resulting into a very pleasant skin without high chances of damage. What are the advices? Unfortunately, as I said already before, there are a few problems. There is no evidence about what is the best therapy. So that means that all the doctors all over the world will have their personal preferences to treat a disease. Also, in some countries, reimbursement can be giving a problem because all the treatments can be very expensive. What are the most used creams? You see here are a lot. I have found in the literature more than 17 different creams for the same disease, but without any evidence, so always was it expert opinion by one doctor. What is currently advised? If treatment of agents is insufficient, you should use topical corticosteroids. And mostly when they are used, you choose one of the 4 classes in increasing strength from 1 to 4. Category 4 is only advised you in very severe diseases, and mostly in the Netherlands we need a dermatologist who give us advices for this. Also on scar management, you see that there are always a combination of pressure with silicone gels and a moisturizer, and also UV protection. Correction will lead to treatment to a good scar maturation. However, hydration by means of moisturizers should be continued, sometimes lifelong. And that's one of the pitfalls that can happen when some people stop using their creams. So as I said, treatment by using moisturizer as initial therapy, you should do it at least until epithelialization, but sometimes longer. The hypothesis of the treatment is that hydration of the epidermis and restoration of the skin barrier will restore balance in the skin and adds to the prevention and treatment of additional normal scarring, especially in patients at high risk of active scar formation. One of the biggest questions is what is the right moisture and what are the components should be there in a creamer. And Klotz et al. Did a survey and came to is the only in his article who mentioned what he found that was very important. It should be conducive to scar maturation. It should be low irritant. It should prevent skin drying and negative effects to the skin barrier function, and it should minimize abundant transepidermal water loss. This is the only time I will mention the name. Alidran is called MMRC, and MMRC stands for Medical Moisture Retention Cream. So the firm tells us that it's able to restore the water balance. It's available on the Dutch market since 2005. And at the time, it is specially used for dermatitis, itching, pain, and treatment of hypertrophic scars. What is MMRC? It's an oil in water emulsion with its most important part, aloe vera. It's made only on Aruba because that's the best temperature and climate to get a very good aloe vera gel. It's also composed with oils and fatty ingredients. And the moisturizing effect is done by the aloe vera and adequate inclusion about the fatty ingredients, and it also includes emollients, so there is water, a better water system inside the skin. Is there a lot of literature? And unfortunately not. There are only two published articles, one of Hoeksema et al. From the University of Ghent, Burning Center, and who looked at the effects of MMRC on burn wounds. And there's a study from Rondas and Scholz about MMRC in especially dry skin dermatitis caused by venous insufficiency. So what are the results of Hoeksema? They did a prospective open controlled comparative trial of which the objective was to compare the inclusive and hydrating properties of MMRC versus 3 forms of silicone gels and 4 silicone sheets. The taped script skin was used as a standardized scar-like model on both forearms of 40 healthy volunteers. And at different time points, transepidermal water loss, as well as the hydration state of the stratum corneum, was measured with a special tool and compared with intact skin and a scare-like control over a 3 to 4 hour period. What were the results? They showed that a hydration cream could provide the same occlusing and hydrating properties as the normally advised fluid silicone gels. It provided a good occlusion and hydration of a period of at least 4 hours. And in the second part, they saw a decrease of the transepidermal water loss and provided a well-balanced hydration state of the skin scar model to near normal values. Rondas and Scholes, they had a case study about 18 patients with venous eczema or venous dermatitis, also another used name. In total, 80 patients with moderate eczema were included. The most important indication were dryness, itching, and/or a flaky skin. And 70 patients were very pleased with the cream because it resulted in a correction of the transepidermal water loss balance. They had as a conclusion that the use of MMRC in patients having an itchy, scary, or dry skin due to underlying chronic venous insufficiency may decrease the topical use of corticosteroids. And based on this study, a lot of clinics in the Netherlands now use at first this type of cream instead of corticosteroids. There are also some presentations done at large symposia, as you can see in these two slides, all with the same result, a good hydration of the skin. Next to this, there are some experiences, case reports. What we can, if you like to have them, you can order them at the company. What is my experience with MMRC is that there is a good pain reduction during wound dressing changes. This is not is just on my own experience, so it needs more research to make it as a standard procedure in wound treatment. There's a very good itching reduction. The conditioning of the skin will raise very good. And now I'm started with treatment of blemishes with and without fungal infection, because that is a big problem in the elderly care facilities, because skin is very vulnerable for this type of disease. What are my conclusions? With increased skin hydration and reducent the property may contribute to preserve an adequate skin barrier function, and that will result in a better quality of life. My take-home messages: several pathologies share the common origin of a disrupted skin barrier, so not only dry skin, but also eczema, psoriasis, and scars. A disrupted skin barrier results in increase of transepidermal water loss. Current guidelines advise moisturizers for improvement of a disrupted skin barrier, and moisturizers should be prescribed based on evidence instead of reasons as reimbursement and personal preferences. A good moisturizer contains 3 components, and good moisture should be conducive to scar maturation below irritant and minimize transepidermal water loss. So MMRC is a good moisturizer. It contains all the 3 components. 2 studies and a lot of presentations show an increase of hygiene and decrease of transepidermal water loss. Its property contribute to an adequate skin barrier, and it helps patients with a disrupted skin. Thank you for your attention.
Thank you, Dr. Martin. It was a nice presentation. Is anyone having any questions? Thank you once again. I think there are no questions.
Okay,
So
thank you. Then I leave the meeting. Bye-bye.
yeah, bye-bye, Dr. Martin. Yeah, so now we will move toward the next presentation. It is from Andras Simonovitz, but as he was busy, so he requested us to like we can go further for the next presentation. So I will request Dr. Nilüfer Ayak from Turkish Thoracic Society, Turkey, to present their presentation. Hello, Dr. Nilüfer. Are you ready to give the presentation? Hello, Dr. Nilüfer.
My name is Oktay Özten. Nilüfer Hanım da burada. Nilüfer Hanım katılmak ister misiniz? Ben başlayayım istersen.
Hello, I am Oktay Özten from Istanbul Metropolitan Municipality, and I will be doing presentation.
Can I know whose presentation are you going to present?
Director of the Directorate of Alms House of the Metropolitan Municipality of Istanbul.
Is this for Dr. Nilüfer Ayak presentation?
Yes.
Okay, Dr.
Evet, okay.
So I'm going to make you the presenter. You can start his presentation.
Thank you.
Thank you.
Ben
Yeah, you are now the presenter. You can share your screen and start your presentation.
Do you see the presentation?
Yeah, it's visible.
Thank you. Okay.
Merhabalar. İstanbul Büyükşehir Belediyesi Yaşlı Bakım Hizmetleri Darlıca Müdürlüğü'nde COVID-19 sürecini nasıl yönettiğimize ilişkin sunumumuz var.
Hello, we have a presentation about the management of the COVID-19 process at the Istanbul Directorate of Alms House.
İstanbul Yaşlı Bakım Vadimiz göründüğü gibi dünyanın en büyük yaşlı bakım hizmetlerinden bir tanesidir.
As you can see here, this alms house is one of the biggest alms houses around the world. Yep.
Yaşlı bakım hizmetlerimizden şu ana kadar kapasitemiz 1066 kişilik, fakat 700 sakinimiz yararlanmaktadır.
We have a capacity of about 1,066 people, but we have around 700 residents.
Sağlık üzerine bütün hizmetler yaşlı bakım vadimizde sürdürülmektedir.
We are giving care and we provide all this support in our nursing home to the elderly people.
Görmüş olduğunuz gibi tesisimiz yukarıdan uzaydan görünümü bu şekildedir.
And this is the bird view of our facilities, as you can see.
Dünyanın en büyük huzur evlerinden bir tanesi olup sadece yaşlı bakım hizmetleri sürdürmemektedir.
It's one of the biggest nursing homes across the world, and we do not only provide elderly care services.
Darülazze Müdürlüğü olarak pandemi sürecini nasıl yönettiğimize ilişkin verilere geçmek istiyorum.
Now I'd like to want to give you some data about how we managed the COVID-19 process at the Istanbul Directorate of Alms House.
COVID-19'nin Wuhan kentinde yayılmaya başladıktan sonra dünyanın bütün ülkelerine bazı yollarla yayılmaya başlamıştı.
Right when the COVID started to spread around the world from the Chinese city of Wuhan,
ülkemizde henüz herhangi bir vaka görünmeden biz tüm önlemlerimizi vaka varmış gibi çalışmaya başladık.
we started to take measures even even when there was no single case in our country, and we started to work on devising our measures.
İstanbul Büyükşehir Belediye Başkanımız Sayın Ekrem İmamoğlu kendi tasarrufları doğrultusunda İstanbul Belediyesi'ne bağlı Sağlık Bilim Kurulu oluşturdu.
The Mayor of Istanbul, Mr. Ekrem İmamoğlu, founded the scientific committee. As per his instruction, a scientific committee in Istanbul was founded, and we made our action plan following its recommendations.
Oluşturmuş olduğu ve hizmete sunmuş olduğu bu bilim kurulu kendi tavsiyelerini her hafta sonu yayınlayarak bizlere ulaştırıyordu.
This scientific committee published its recommendations every weekend.
Ulaştırmış oldukları bu verilerle beraber İstanbul Büyükşehir Belediyesi'nin yaşlı bakım hizmetlerinde bir eylem planı düzenledik.
And with the data we took from these recommendations, we devised an action plan at the Istanbul Metropolitan Municipality.
O sıralarda dünyanın her tarafında, Avrupa'da, İtalya'da, İspanya'da, Fransa'da birçok huzur evinde yaşlıların terk edildiğini veyahut da ölüm oranlarının arttığını gördük.
And we saw that at that period around the world, in countries such as Italy, Spain, or France, in many nursing homes, people were left to be alone, the elderly people.
Tünelin sonu gözükmüyordu ve bizler ne yapacağımız noktasında bilim kuruluyla beraber güzel bir yol alıyorduk.
We weren't able to see the light at the end of the tunnel, but we worked with the scientific committee on that regard.
Dünyada çok sayıda COVID-19 salgını, dünyada çok sayıda ülkede COVID-19'un yayıldığını görünce yapmış olduğumuz eylem planını 1 Mart tarihinde yürürlüğe koyduk.
And when we saw that COVID-19 was fast spreading in all the countries around the world, we devised and put into implementation our action plan on the 1st of March 2020.
Ve bunu 12 Ekim 2020 tarihine kadar 0 vakayla getirebildik.
That's why actually because of this success, until 12th of October 2020, there was no single case in our almshouse of COVID-19.
Az sonra sizlere paylaşacağım eylem planımızı nasıl yürüttüğümüze gelirsek eğer,
Now I'd like to share with you briefly the details of our action plan.
öncelikle COVID-19'dan etkilenecek olan yaşlılarımızın birer analizini çıkardık. Bu analizleri yaparken doktorlarımız çok titizlikle davrandık. COVID-19'dan etkileneceklerin listelerini hazırladılar.
First of all, we started with analyzing our elderly, and our doctors took really great care in terms of devising and giving every detail about our elderly people.
Bu kronik rahatsızlıkları olan risk grubunun ayrı alanlarda kalmalarını sağladık.
And we determined people who had accompanying diseases and who are in the risk group, so we put them in other areas, separated them from the rest of the group.
Gördüğünüz gibi yaş grubumuzun ortalaması 70 civarlarında bulunmakta olup ve COVID-19'a maruz kalınması durumunda bütün yaşlı bakım hizmeti sürdürdüğümüz 700'e yakın sakinin etkileneceği göz önünde bulundurursak yapmış olduğumuz eylem planının ne kadar önemli olduğunu ve sıkı tedbirlerle uygulanması gerektiğini uygulamaya koyduk.
The importance of our action plan and the importance to very properly implementing it becomes very clear if we take into account that the general age average at our almshouse is 70.2. So these people being exposed to COVID was a high risk, but we took our action plan and all the measures in that regard.
COVID-19 dünyada yayılmaya ve sınırların ötesine geçmeye başladığı andan itibaren yapmış olduğumuz en büyük hizmetlerden başlangıcında Coalesce yani yaşlı bakım hizmetleri olan kısma giriş çıkışları kapattık.
And as soon as the virus started to spread around the world and detected within the Turkish borders, the very first measure that we took was to close entrances and exits from and to our almshouse.
COVID-19'un belirtileri arasında yer alan yüksek ateş, sıkıntılı nefes alıp verme, yaşlıların en çok yaşamış oldukları ateş yükselmesi veyahut da bir takım rahatsızlıkların oluşmasına sebep olan tüm etmenleri gözeterek izolasyon alanları oluşturduk.
And we created isolation spaces taking into account some residents who showed some symptoms, such as high fever or respiratory distress. So we put them into isolation areas.
Biz yaşlı bakım hizmetleri sürdürürken bütün bu hizmetlerin herkese eşit miktarda yansımasını sağlamak amaçlı eylem planımızı önce eğitimlerle başladık.
So we started off with offering trainings so that everyone at our almshouse would be treated equally in terms of implementing the action plan.
Bu eğitimlerimizi verirken gıda mühendislerimiz, diyetisyenlerimiz, hekimlerimiz, bütün çalışanlarımız ve sakinlerle görüşmeleri yaptılar. Ara öğün ve ana öğünlerine takviyeli minerallerle güçlendirdik.
Before and during these trainings, food engineers, dietitians, and doctors, we worked with them, we consulted them, and they worked with our residents. So we even increased the diversity of food, both for in-between snack times and during main meals.
Kamu özel sektör kuruluşlarından bir tanesi olan İstanbul Büyükşehir Belediyesi, Büyükşehir Belediyesi bünyesindeki tüm hizmetler eşit olarak sürdürüldüğü için yaşlı bakım hizmetlerimizdeki tüm COVID-19 KKT yani bunu nasıl çevirirsiniz bilemiyorum ama yani eldiven, maske, gözlük gibi koruyucu güvenlik talimatlarına uygun her şeyi sağladık.
And we provided each and every equipment such as gloves or masks or any other protective equipment in all the stages of the elderly care in the fight against COVID at the Directorate of Almshouse as a public institution operating under the Istanbul Metropolitan Municipality.
Hijyen kuralları noktasında sağlamış olduğumuz bu çalışma bizim etkin, verimli, disiplinli bir şekilde eylem planına sabit kalmalarını düzenledik.
So actually this measures about hygiene that we took shows how effective and with discipline our action plan was implemented.
İlk eylem planlarımızdan bir tanesi olan sakin kabulünü durdurduk. Yani o saatten sonra COVID-19 yayılmaya başladığı andan itibaren sakin kabulünü durdurduk.
And another thing that we did within the framework of our action plan was to stop admittance of new elderly residents at our almshouse.
İçerideki yaşlı bakım hizmeti sürdürmüş olduğumuz sakinlerin yaşam kalitelerini arttırarak dışarı İstanbul'a çıkışlarını engelledik.
And we increased the living standards of our already existing residents at the almshouse so that they wouldn't need to go outside the premises around Istanbul to meet their needs.
COVID-19'un etkilerini azaltmak amaçlı kurum içerisinde izolasyon alanları oluşturduk. Hastaneye gidip ve geri gelen sakinlerimizin içeride yatılı kalan sakinlerle olan diyaloglarını kestik.
In order to reduce the risk of transmission of COVID-19, we created isolation spaces and we separated people who had been transferred to the hospital and who came back to the almshouse so that they would not contact each other.
İçerideki doktor sayısını arttırdık.
We increased the number of doctors.
Böylece hastanelere sevki azalttık.
We increased the number of doctors who serve within our premises so that we would actually decrease the number of cases referred to the hospital.
WhatsApp grupları kurarak 24 saat eylem planımızı arkadaşlarımızla paylaştık.
We shared our action plan 24 hours by way of communication on WhatsApp groups.
Dezavantajlı, kronik rahatsızlığı olan tüm çalışanlarımızı izne çıkardık.
And all staff members who had chronic diseases who are disadvantaged were put on administrative leave.
Bu süre içerisinde idare işlemleri sürdüren tüm personelimizin toplu taşımalarını kendi araçlarımızla sağladık.
And all the administrative staff members were brought from and to the working place at our almshouse by free shuttle bus service.
Kurum içerisindeki tüm sakinlerin tarafımıza fizik tedavi merkezinin hizmetlerini sağlayarak fizik tedaviden daha çok yararlanmalarını sağladık.
And we allowed the residents of our almshouse to have access to the services of the physiotherapy center in-house.
Sürecin 8 Nisan'ına geldiğimizde yapmamız gereken eylem planını bir kat daha arttırdık. Bu süreçte dünya tarihinde olmayan bir olayla karşılaştık.
And when we arrived at April 8th, we actually doubled our efforts.
Covid-19 Türkiye'ye gelmiş ve yayılmaya başlamıştı.
By that time, COVID-19 had already reached Turkey and started spreading.
İlk eylem planlarımızın bir tanesi olan tüm çalışanlarımız, sağlık çalışanlarımızı PCR testinden geçirerek 15 günlük yatılı periyotlarla kurum içerisinde yatılı kalmaya sağladık.
First of all, we applied COVID-19 PCR test to all staff members, and we started working on shifts of 15 days.
Tam bir yıldır bu hizmeti 15 günlük periyotlarla sürdürmekteyiz.
For the past 1 year, we have been continuing this system of shifts, 15 days working at the almshouse and 15 days resting at home.
Bu süreç bize ne kadar fayda sağladığı PCR test sonuçlarının pozitif çıkanların içeri almayarak gördük. Pozitifleri içeri almıyoruz.
And this process has contributed to our efforts so much that when the PCR test results was positive, we didn't admit these people at the almshouse.
Bu 380 personelimiz içeride yatılı kaldı. Otel kısmı ise tüm donanımlara sahiptir.
And these staff members of about 380, when they stay at the almshouse in the hotel section of it, they can make all their needs met because they have all the facilities.
Evet. Sakin olan sakinlerimizin, yaşlı hizmeti sunmuş olduğumuz sakinlerimizin kendi içeride kaldıkları süre içerisindeki yapacakları harcamalarda kendilerine ödenen paraları dışarı gidip bankadan çekmek yerine ayaklarına getirdik.
And in order to protect our residents who were withdrawing their salaries from ATMs, we stopped this system, and so we put an official person who would go get their salaries and give them their salaries, so they don't need to go to the ATM machine.
Bu süre içerisinde yapmış olduğumuz çalışmalardan bir tanesi de devamlı kurum içerisinde dezenfektan olaylarının hiç durmadan devam etmesiydi.
And one of the other measures that we took during this whole process was to constantly disinfect all the areas.
Yaşam standartlarını kaliteli bir şekilde sürdürmeleri için milli bayramlarımız olan 23 Nisan, 19 Mayıs, 30 Ağustos Zafer Bayramlarını günün anlam ve önemlerine bağlı kalarak içeride kutladık.
And in order to sustain the high life standards of our residents, we even celebrated our national holidays within the premises.
Sosyal yaşantı alanlarında İstanbul'da sokağa çıkma kısıtlamalarını değerlendirerek kendilerine İstanbul turları yaptırdık.
And when there was limitation for the general public to go outside in Istanbul when there was lockdown, we took this opportunity to offer our residents some socialization activities. So we took them outdoors.
Eylem planlarımızdan biri olan vardiyeli çalışma sistemini düzenli olarak yaptığımız periyotlarla devam ettiriyoruz.
And we are continuing to this day the system of working on shifts.
Covid-19'un belirtilerini önceden gözetecek şekilde kurum personelinin giriş çıkışlarında iş sırasında 3 kez ateş takibi yapılmaları halen devam etmektedir.
And we are still taking the temperatures of our staff members 3 times per day in order to determine the symptoms, if any, to actually know them in advance.
Kampüs içerisinde sürekli devamlı maske kullanımını zorunlu hale getirdik.
We have made it an obligation to always wear a mask within our campuses.
Nisan ayında almış olduğumuz bu uygulama bir yıldır sürekli bir şekilde devam etmektedir.
It has been continuing since the month of April.
İdare ve huzur evleri arasında geçişler sonlandırılmıştır. Yani bizim yaşam alanlarını, sakinlerin yaşamış olduğu alan ile şu anda bulunmuş olduğumuz idari kısım arasında bir periyot oluşturarak geçişleri kapattık.
So we stopped people from moving between the administrative buildings where we are situated right now, giving this presentation, and the nursing home areas.
Bu süreçte, özellikle bu süreçte sakinlerin yaşayabilecekleri sosyolojik ve psikolojik durumları göz ederek alanda sosyologların ve psikologların daha etkin şekilde çalışmaları için onlara birim oluşturdu.
And we created a unit for sociologists and psychologists so that they would work ever more closely with people, especially during this period where people can run into social or psychological problems.
Bu süreç içerisinde içerideki sakinlerimizin ailelerine görüşmelerine WhatsApp ve Zoom üzerinden bir takım sosyal iletişim üzerlerinden sağladık.
And we facilitated the communication of our residents with their relatives and family members by way of allowing them to have video calls on WhatsApp or Zoom.
Kurum dışından idari manada kurum içerisinde herhangi bir ziyaret olmaması çok sayıda COVID'in önlenmesine sebebiyet vermiştir.
And since there was no visit from outside the facilities into our facilities, we prevented the cases of COVID.
Sonuç itibariyle pandemi süreci çok yönlü ve sürdürülebilir mücadeleyle Darülfünun Müdürlüğü içerisinde sürdürülebilir bir yer almıştır.
In conclusion, I'd like to say that we took our fight against COVID, we struggled against it on a multifaceted manner and sustainable manner.
Hızlı ve zamanında almış olduğumuz kararlar ile
With the decisions we have taken in a fast manner,
sürdürmüş olduğumuz disiplin sonucunda
and thanks to the discipline that we followed,
bu başarıyı elde ettik.
we actually were very successful in our fight against COVID.
Kamu kaynaklarının etkin kullanımı noktasında sürdürmüş olduğumuz bu hizmet
And we are actually providing this service by effectively using the public resources.
tüm ülkede 15 günlük, tüm ülkedeki diğer huzur evlerinde 15 günlük periyoda geçmesini sağladı.
And actually it served as an example for other almshouses and elderly care homes across the country in terms of our shift system, 15 days.
Bizden daha sonra dünya çapında her tarafta bizi arayarak bu konunun nasıl işlediğimizi sorgulayan huzur evleriyle eylem planımızı paylaştık.
And we have shared our action plan with the nursing homes across the world who reached out to us to actually learn the details of our action plan and learn the details of the shift system, 15 days at home and 15 days at work.
Asıl vurgulamamız gereken en önemli unsur ise kamu hizmetinin sürdürülebilmesi için bu tür hizmetlerin yani bakım evlerinin, huzur evlerinin kamusal ve kurumsal yapılarla sürdürüldüğü zaman nasıl güzel sonuçlar doğurduğunu ortaya çıkarıyor.
And the main takeaway from my presentation would be that when you actually when you lead this process by effectively using the public resources while you are actually serving in a public manner, it will cause really great outcomes.
Görmüş olduğunuz tablodaki gibi yeni vaka 2 Mayıs itibariyle, şu paylaşacağım 2 Mayıs itibariyle 808 bin dünya çapında vaka olduğu bir ortamda Türkiye ise ilk 100 bin içerisinde yer almaktadır.
And as you can see on the screen, we have
ilk 100 bin içerisinde 3000 vaka olduğu gözlemlenmektedir.
As you can see on the screen, per day globally, new cases are around 808,000, and in Turkey, per 100,000 people, we have more than 3,000 cases.
Şu an itibariyle dünyada 3 milyon 186.538 ölümün olduğu ortamda 1 milyar aşının yeterli olmadığı gözlemlenmektedir.
And across the world, we have around 3 million deaths. And so vaccination of around 1 billion people is not enough.
Görmüş olduğunuz gibi COVID-19'un süreçleri aşamasında yeniden ölüm oranları dünya çapında artmaya geçmiştir.
And you see on this graph that the rate of death is again increasing across the world due to COVID-19.
Bu durum ağır vaka ile eşit orantılı gitmektedir.
And this situation is going in line with serious patients.
Dünyada bu oranların arttığı 19-25 Aralık'ta ilk 100 binde olan ülkeleri görmektesiniz.
And here on this map you can see the time period between 19 to 25 April, COVID-19 cases per 100,000 people.
Sonuç olarak
So in conclusion,
İstanbul Büyükşehir Belediyesi ve onun bilim kurulu ışığında yapmış olduğumuz bu eylem planıyla beraber tüm çalışma arkadaşlarımız ve doktorlarımızla beraber sonuca başarı ve emin adımlarla ilerlemekteyiz.
I'd like to say that in the light of the action plan that we devised after the recommendation of the scientific committee of the Istanbul Metropolitan Municipality, we worked really closely, both the staff members and with our doctors. So we are really successful in our fight against COVID.
Dünyanın en büyük huzur evlerinin bir tanesinde bu kadar büyük başarıyı elde etmek için bana ve ekibime bu onuru yaşatan tüm çalışma arkadaşlarıma, başta bizim önümüze aydınlatan, önümüze ışık tutan ve bu tünelin ucunu görmemizi, tünelin ucu görünmeden bize moral vererek bilim kurulunu oluşturan Büyükşehir Belediye Başkanımız Sayın Ekrem İmamoğlu'na, onun önderliğinde sürdürmüş oldukları bilim kuruluna şükranlarımızı sunarız.
So actually, I would like to thank all the members of my team and people who have worked closely with us, because this is not easy to achieve in one of the biggest almshouses and elderly care homes in the world. So I would like to primarily thank Mr. Mayor Ekrem İmamoğlu, the mayor of Istanbul Metropolitan Municipality, who has allowed us to see the light at the end of the tunnel and who created the Scientific Committee of Istanbul.
Ve bize bu yayını yapmamızda ve bizim bu yayınımızda bize organizasyonda emeği geçen herkese, başta doktorumuz Sayın Nilüfer Hanım'a ve bütün bilim kurulu üyelerine ayrıca tek tek şükranlarımızı sunarız.
And I would like to lastly thank all people who have made this organization possible and gave us the opportunity to present. And I'd like to thank our doctor, Ms. Nilüfer Aykaç, and all the other scientific committee members for all their efforts.
Söylemek istediğim şey varsa sorumuz bitmiştir.
And our presentation has come to an end. If Ms. Nilüfer wants to say anything, she's welcome to do so. Otherwise, thank you for listening.
Thank you, Dr. Okti. It was a nice presentation. Is anyone having any questions? Okay, thank you once again. So we will move forward for the next presentation. It is from Epimenandis Doxakis from Biomedical Research Foundation of the Academy of Athens, Greece. Hello, Dr. Epimenandis.
Hi.
Are you ready to give the presentation?
I am ready, yes. I can hear you well.
Yeah, I'm going to make you the presenter.
Okay.
Yeah, so now you can share your screen and start your presentation.
Buradan çıkabiliyoruz.
Yeah, your screen is visible now. You can start your presentation.
Hang on, I can't see my PowerPoint.
Okay.
Okay. Can you see the presentation?
No, doctor, it's not visible.
All right, okay. Well, let me try again. Can you see the presentation now?
Yeah, it's now visible. I think it's not loaded. Yeah, it's perfect.
Okay. All right. So there are no conflicts of interest. Do something here. Right. So I would like to thank the organizers, particularly Brian, for giving me the opportunity to present my work here in this webinar. As it will become shortly clear, there is an urgent need to identify novel biomarkers, prognostic and diagnostic biomarkers for Parkinson's disease. MicroRNAs have been put forward as promising biomarkers for PD. Now, something is wrong.
What happened, doctor? Is there any problem?
I can't change the slide.
Yeah, you can scroll your mouse.
Okay, so
no, your presentation is not visible, doctor.
All right, okay.
Yeah, now it's visible.
All right. Okay. Okay, so I'm going to give the, can you see that? Can you see
Yeah,
the
yeah, it's visible.
I'm going to give a brief introduction to Parkinson's disease. So it is a progressive neurodegenerative disease. It affects over 7 million people worldwide, and it is expected to, but this number will double by the age of 2040. It is predominantly an older people disease. So as you can see, it's only 1 in 100,000 under the age of 50 that develop Parkinson's disease, while people who are over 60 have 1 in 100 chance of developing Parkinson's disease. It is an idiopathic disease, and only 10% of it is familial. And you can see here some of the genes that are implicated in Parkinson's disease. Parkinson's disease is best known for its motor symptoms, like resting tremor, bradykinesia, muscle rigidity, and postural instability. But there are a lot of non-motor symptoms accompanying this disease, like neuropsychiatric symptoms, autonomic, sensory sleep disturbances, and sensory symptoms. The neuropathological hallmark of Parkinson's disease is the death of dopaminergic neurons in the substantia nigra in the midbrain, and also the presence of inclusions rich in alpha-synuclein, phosphorylated alpha-synuclein, which are called Lewy bodies in remaining living neurons in substantia nigra and elsewhere. Lewy body pathology can also be observed in the cortex, in the enteric plexus, and olfactory epithelium. There is no therapy for Parkinson's disease, and current treatments are only symptomatic. You can see here the different types of treatment. These include levodopa, dopamine agonists, monoamine oxidase inhibitors, methyltransferase inhibitors, anticholinergics, and when medication doesn't work, then surgery can be employed, such as deep brain stimulation, and less often lesioning the thalamus or subthalamus, and also stem cell therapies. Nevertheless, there is also a need for a number of other specialists to help with the day-to-day care of these patients, which include physiotherapists, speech therapists, occupational therapists, and dietitians. Okay, so what constitutes a good biomarker? So good biomarkers should be consistent across gender and ethnic groups. It should be sensitive and specific for the disease. It should be obtained with minimally invasive procedures. It should be stable in biofluids, and then its detection and quantification should be safe, robust, rapid, simple, accurate, reproducible, and inexpensive. With respect to Parkinson's disease, we currently use rating scales, which are basically questionnaires for these patients, and include probe motor and non-motor symptoms. The problem with the rating scales is that they are often subjective and are influenced by periodic fluctuations in symptoms and also some effective symptomatic therapies. Then there is also imaging, such as a DAT SPECT, which really works very well. It can provide a quantifiable measure of the disease progression, but it is limited by practicality and costs. And there are also some protein biomarkers which are now are examined, but unfortunately they don't seem to be very reproducible or do not reflect disease progression. MicroRNAs have come up as really very good biomarkers for a number of diseases, including cancer and other neurological diseases. Because they are small, just a small background, so they are small, non-coding, double-stranded RNAs. They control the expression of about, on average, of about 200 mRNAs by base pairing on the free UTR of these mRNAs. It is expected to regulate up to 6% of the protein. We have more than 2,500 microRNAs in humans, and these microRNAs are either ubiquitously expressed or we have tissue or cell-specific expression pattern. Importantly, they are secreted by all cells of the body and can be found in all body fluids, including saliva, serum, plasma, CSF, and tears. There's a little variation between sexes, age, or time of sampling. And the important thing is that we can get clear-cut interpretation. So levels correlate, microRNA levels correlate with activity, which is not the case with proteins, because as you know, proteins are modified by methylations, glycosylations, acetylations, and so forth. So levels are not really correlated with activity in case of proteins. And another very important characteristics of microRNAs is that they can be quantified by routine and fast methods that can be now found in most research settings and hospitals. So this is the workflow of the work I did, and this work can be found in the research review paper last year. So basically, what I did is that I did a systematic literature review to identify microRNAs that are consistently deregulated between studies. Sorry, I missed to say that, but there have been about 24 different studies on microRNAs in PD, but results have been rather inconsistent. So I decided to basically review all these data, integrate the data, and identify which microRNAs are deregulated. As you can see here, there were 81 uniquely deregulated microRNAs, of which 25 of these were seen in at least 2 biomarker studies. Now, what I did next was actually try to understand the biological role of these microRNAs and study the chromosomal distribution, the transcription factors that regulate these 25 microRNAs, as well as identify the targets of these microRNAs and the role of these targets in Parkinson's disease pathogenesis. This is an overview of the different studies that are out there. So most of the studies are discovery studies. They do not have a validation component. The number of PD subjects varied, and only 50% of the studies actually had more than 50 PD patients, which I think is the absolute minimum to actually make sense of microRNA data, as microRNA changes are not really dramatic. The fluid fractions that were used in these studies were mostly from blood. The RNA extraction method varied significantly. The microRNA detection method was qPCR, and there was a very big variation in the reference genes that was used for normalization, and I think this was one of the issues that led to not identifying very similar microRNAs. This is a picture of the different of the 81 microRNAs that were identified. As you can see here, this is the number of times, number of reports these microRNAs were identified. You can see here that there were 25 microRNAs that were reported in 2+ studies, 11 microRNAs that were reported in 3+ studies, and 4 microRNAs that were reported in 4+ studies. The interesting thing was that many of these microRNAs are really relevant for brain function and also Parkinson's disease pathogenesis. So for instance, 124 microRNAs, the most highly expressed microRNA in the brain, and regulates neuron cell specification and differentiation, as well as it is protective against Parkinson's disease oxidants. MicroRNA 128 is a microRNA that regulates ion channels that are important for motor activity. 132 regulates transcription factor NUC1, which is important for dopaminergic neuron specification, but also it's important for memory formation and retention. MicroRNA 19 is very important for the migration of newborn neurons generated in the adult, and microRNA 29, which is actually downregulated in Parkinson's disease, regulates methylation, and this is kind of fits with what Laura presented earlier on with methylation studies. So it appears that microRNA levels go down, so methylation goes up in the elderly as well as in Parkinson's disease. So the take-home message is that we now have a number of microRNAs that have been shown in these 24 studies, and we can now probe with more studies to see if these microRNAs are consistently deregulated. Nevertheless, I moved on to try to get some information about the role of these microRNAs. So as I said, I look into the transcripts that are regulated by these microRNA, and you can see here the different pathways that are regulated by the microRNA targets. And this is the FOXO signaling pathway, prolactin signaling pathway, TNF, and RB signaling pathway. These are all very critical pathways for Parkinson's disease. FOXO signaling, FOXO transcription factors are the transcription factors that control prolongevity. They control the expression of antioxidants and autophagy genes. Prolactin has not been studied very much, prolactin signaling, but it has not been studied very much in the brain and also in Parkinson's disease, but it is well known that when dopamine levels go down, like in Parkinson's disease, prolactin expression is significantly increased. TNF signaling is another pathway that we know well that it is activated in the brain of Parkinson's disease patients. So there is a microglia activation and also an infiltration of T cell lymphocytes in the substantia nigra of these patients. And last is RB signaling. Again, RB signaling is very important for the survival of dopaminergic neurons. Now, what was really striking to me was then that when I looked into the biological processes that are controlled by the mRNAs that are targeted by the deregulated microRNAs, was that most of the categories were involving transcription. So that means that there is not only deregulation at the microRNA translation level by microRNA deregulation, but there exists also a second wave of en masse regulation involving transcriptional-wide changes. So obviously, when I also look into the molecular functions of the microRNA targets, again, the top 8 categories brought out transcription. So most the targets of the deregulated microRNAs are transcriptional proteins, transcription factors, and again we look into the place where these these proteins are resident, and it's obviously the nucleus and the nucleoplasm. So these are the transcription factors that appear to be regulated by the deregulated microRNAs. And I then conducted an interpro analysis to find the type of transcription factors they belong to. And you can see these are nuclear hormone receptor type or GATA type. And again, this is something that rings bells to PD researchers, because GATA transcription factors control the balance between inhibitory and excitatory neurons in the midbrain, and also control the expression of alpha-synuclein, which is the premier gene, the gene that is thought to be implicated in Parkinson's disease pathogenesis. Moreover, the nuclear hormone receptors include the BGC1A transcription factor, which is well known to control the transcription of all mitochondrial genes, and also NUR1, which is the gene responsible for the dopaminergic neuron specification. So, so this is quite an interesting feat here. Then what I did was try to look on the chromosomal localization of the deregulated microRNAs in Parkinson's disease. And strikingly, as you can see, the long arm of chromosome 14 houses 9 out of the 25 deregulated microRNAs. There are also some other classes. You can see here one cluster and another cluster and another cluster here. But this is really, really striking, because it indicates that there is some transcriptional control there is misregulation of a transcription factor that regulates most of these microRNAs. Another interesting observation was that this site, actually, I was really struck by the number of microRNAs that are located in this site. So I searched the PubMed for this site, and it was striking to find out that this site houses a microdeletion, and there are 6 reports in PubMed showing that people who have got a microdeletion in this site display motor delay, hypotonia, feeding problems, which are typical for Parkinson's disease patients. And this microdeletion is thought to occur because of some expanded TGG repeats that are flanking these microdeletion site, which is about 1 million bases in length. And another interesting observation was that within the same region of these microRNAs, there is a transcription factor which is called the yin-yang 1. And this transcription factor appears to regulate the transcription of these other microRNAs that I have them here in circle. So it seems that this particular region have importance in Parkinson's disease. So what I did next was to use an experimental database, the Transmir V2 database, which holds experimental data, so it's not prediction data, to look on the transcription factors that are regulated these microRNAs. And you can see here that there are the different transcription factors that came up. 15 out of the 25 microRNAs are regulated by CREB1, which is the most important transcription factor for brain development. And in fact, all 9 of the microRNAs that are found in the long arm of chromosome 14. The point I want to make here is that it is possible that somatic mosaicism that occurs at chromosome 14 due to this high genomic instability may actually be responsible for Parkinson's disease. And I think it will be very important for people like maybe Laura, who is doing methylation studies and maybe people who deal with chromosomal abnormalities to check whether there is mosaicism on this region for PD patients, in PD patients, which may explain why we observe these phenotypes. And with that, I would like to acknowledge my collaborators who provide samples for our biomarker studies. I didn't say that we have already conducted two large biomarker studies, and now we are in the process of conducting a third biomarker studies, including other Parkinsonism symptoms like syndromes like MSA and PSP. And also I would like to thank the funding bodies, the Fox Foundation in New York and the Greek Secretariat for Research and Development in Greece, who were generous in providing us funds to conduct our research. And with that, I would like to thank you for your attention.
Thank you, Dr. Epimenandis. It was a nice presentation. Is anyone having any questions? Okay, thank you once again. So we move into the next presentation. It is from Dr. Lev Salnikov from SB Enzyme US LLC USA. Hello, Dr. Lev. I'm sorry, your voice is not audible.
Oh.
Yeah, now it's audible. Yeah. I'm going to make you the presenter. Yeah, now you can share your screen and start your presentation.
Okay, thank you. Thank you very much for this opportunity. And I don't present any slides. It's just a presentation about basic biology of the aging, and it's look like I'm only who present these fundamental questions. And for now, as literature says us, we have about 300 theories of aging, and theory, it's important because it's lead to some direction on the research, and then we could use it in the practice. So what we present at the work, that's showing that most importantly in aging process is function of two portions of our genome. Any multicellular body get two types of genome. One we inherit from our one unicell predecessors, and that portion we name housekeeping genome. That's very well known. It's part of genome which not changing through the evolution practically, and like what we have in each cell, biosynthetical possibility or mitochondrial and so on. So what we have of eukaryotes, we have in each our cell. The other portion of genome is a portion which is a field for evolution, all of process which lead to highly specialization cells and tissue played on that portion, integrative portion of our genome. And their relation, their function between two of them, it's a major factor, major. Because if we look through aging process, so we can see that sometimes it's very difficult to say what is cause of all process, what very well of you know, and if it cause of it, of aging itself, or it just consequences of some other process. In our work, we showed that in time of developing, in time of ontogenetic process, the activity of integrative part increase constantly, and this could be primary cause of other aging process, because all what we can do, all our adaptation in any cell, it depends on housekeeping genes. If and what's going on, we start to take the all material from these housekeeping genes to highly specialized genes. So that could be cause of declining ability of our cells to be adaptive, to do the process which all know like regeneration and reparation, and that's most important of all biological process. So, for example, now the preliminary result of I can present you that if we divided genes in different species, we will see close same picture. For example, in methylation process, our analysis showing that methylation of housekeeping gene, it's going higher than methylation on many of integrative part, which mean that ontogenesis as a program needed to really increase highly specialized part of the body, but that goes with the cost. It's actually decline housekeeping activity, and then it's lead to many things which you well know. We could try and we will in our further research to do to create more specific ratio between activity of housekeeping genes and integrative genes in any multicellular body, and that's give us not only the very interesting and very basic marker which could showing what actually any intervention, if you try to do some geroprotection, if it work, if it really help to revive declining housekeeping ability in multicellular genome. And what's important that if you have this very basic tools, very basic instrument which allowed us to see, we could choose different combination, different different products to see how they actually influence on this ratio between housekeeping and integrative genes. For guys, for them who interested on this, you can see 2 articles in a journal, Frontiers in Aging. You can find those articles and you can see all details on it, what was done and how we describe all those things. It's just short presentation of this quite new direction in biology, in major principles in biology of aging. And that's all, and thank you for your attention.
Yeah, hello, Dr. Lev.
Yes, I'm here.
Okay.
Yeah, that's all my presentation, and if I would love to answer on any questions.
Okay, thank you, Dr. Lev, for your information. Is anyone having any questions? Okay, thank you once again. I think there are no questions, so we can move towards the next presentation. It is from Eckler Jacqua from Loma Linda University Health, USA. Hello, Dr. Eckler.
Hi,
Hello, good morning.
good morning.
So are you ready with your presentation?
Yes,
Yeah, so I'm going to make you the presenter.
okay.
Done. So now you can share your screen and start your presentation. You will be having a screen button, third button.
Okay.
Yeah, you click on that.
Yeah, still not able to.
Do you find 4 buttons at the bottom of your screen? One is mic, camera, screen, and leave button.
The
So click
screen
on
is still
screen button.
the screen is still closed.
So it will. Is there any permissions it is asking for?
Go to meetings need to use accessibility features. So it's like I open my system preferences.
Is it asking for any permissions?
Yeah.
I think you need to give the permission to access your screen.
Yeah, which I did. Yeah, it still doesn't allow me. If I forward my presentation, can you share the screen for me?
Yeah, it's done. So I will share you my mail ID. You can send your presentation to me. I will pray on your behalf and you can start explaining to that.
Okay. So
I'm
for
just sending you my mail
what
ID.
email?
Just yeah, I'm writing in the chat box.
Okay.
Yeah, I sent you on chat.
Okay. Okay, I just sent you.
Yeah, I'm just checking it. Yeah, received. Just a minute. I'm going to download it. So I will be sharing my screen. You can let me know when I have to change the slide.
Okay.
Yeah. Yeah. Is it visible?
Yeah. So just press for the presentation. Yeah.
Yeah.
Great.
Yeah.
Okay, great. Yeah, you can. I have a lot of animations, so you can.
Okay.
Yes, perfect. You can keep going. Yes. Good morning or good afternoon, everyone. My name is Eclair Jakeway. I am a geriatrician. I'm also family medicine at Loma Linda University. And today my topic is a little different. It's not like pure research as we are listening the whole day, but it's about reimbursement. So us geriatricians, how can we make sure we have how to optimize our reimbursement from Medicare? So this applies more for the US, but it's just like a talk about things that we can document and be and then we're going to be able to get everything that we are doing for our patients. So the title is Optimize Your Documentation to Improve Medicare Reimbursement. Next, please. Next. So I don't have any disclosures. Next. Learning objectives. So my goal is to define the different visit types in the geriatric population, review required elements for a cognitive assessment visit, and also how to maximize and document for the preventive services that we offer for our elderly patients. Next. Next. Thank you. So like I was telling you guys, there's nothing more frustrating than just not getting the credit. You can go back. They're not getting the credit for the work that we perform, right? So sometimes with our older patients, we spend more time and the reimbursement ended up being the same. So we are providing way more services, way more counseling for them and advising them. But depending on what we document, what we write in our chart is just not going to be recognized. So this happens more with the preventive medicine. So we talk with our geriatric patients about preventive medicine all the time. But if we don't document that right, nobody's going to know that we did it. Next. So older adults. So you can keep next. Yes. So the geriatric patients, they are way more complex, right? So they have so many diseases, polypharmacy, lack of social support. So we spend way more time with these patients, right? So we address all these complaints. We try to address in one visit, like 15, 20 minutes. If we are lucky, we have 30 minutes for new patients, but it's hard, you know. So we need to know how to document what was done. Next. And you know, documenting and billing appropriately. We don't want to under or overcode. And our visits with our older adult patients, they need to be a cost effective endeavor, right? So we need to make sure that all the time, all that counseling we are giving is going to be recognized. Next. Okay, next. Good. So basically, we have, like, I'll say, two main visits, which is the annual wellness visit, which we call AWV, which, as the name say, we do once a year. The standard office visits that they come for complaints or follow-ups. And we have the cognitive assessment visit. The first two are the main ones, and the cognitive assessment visit is something that we can do it by itself. Here at Loma Linda, we have a clinic that we just do a cognitive assessment visit, or we can do along with the annual wellness visit. Unfortunately, we cannot do the cognitive assessment visit with a standard office visit. One, because we don't have enough time, but the Medicare billing, they don't allow it, because the cognitive assessment visit, usually the RVUs, the amount that they pay is higher. So you ended up using that visit instead of a standard office visit. So you cannot do it together, but you can do it at annual wellness visit if you want. Next. And the wellness visit, you can go back, sorry. And the wellness visit, you can also do the cognitive assessment visit together, like I said, and also you can do with a standard visit. If the patient comes with complaints, with issues like a knee pain, shoulder pain, you can split bill. You can do your annual wellness visit and also your standard visit together. So you ended up getting the RVUs for both visits. Next. So preventive services. So what I'm talking about preventive services. So this graph is a very nice one that shows like things that I'm sure we all counsel, we all advise our patients, we all talk with our patients about it, but we don't document. So for example, cognitive assessment, just the assessment, right? Like just asking about the cognitive assessment, just assess, is something that you have just a visit for that, or you can do with a wellness visit. The advanced care planning, same thing. If we talk about the post, if we talk about advanced directives, is something that we can be. We cannot be on the cognitive assessment visit because it's already part of the template, but we can add this RVU on the wellness and the standard office visit. Same thing with depression screening, smoking cessation, alcohol, as long as we know how to document. And I'm going to go to some examples in the end. The cognitive assessment is a little tricky because on your wellness visit, you have the screening questions, but if you're going to do the full cognitive assessment to bill for both, then you spend way more time. So I don't think we have enough time to do both. That's why I rarely see, but it's something that we can do. Next. Okay, so what is the annual wellness visit? So it's a physical exam. So what are you talking about? Standard office visit and annual wellness visit is not the same. So actually it's not. So the AWV that we call is just an incentive visit from Medicare. So it's not a physical. So a lot of people, they confuse that. So it's a health risk assessment. So you're gonna go to the preventive services, vaccinations, all the screening for their age, and a personalized prevention plan. Basically, that's what it is. And there is a template for that. So there is a few things that you need to ask, but it's not a physical. So if the patient comes for an AWV and they have complaints about like, I want to talk about my shoulder pain, my knee pain, my wrist pain, then you can split bill. You can bill for 2 visits as long as you tell the patient, that you disclose that to the patient, because you are doing 2 different things. You are addressing the issue, and also you are doing your annual wellness visit, which is your incentive, your prevention plan, your incentive visit. So there is a few types. So the welcome to Medicare is the same as the initial preventive physical examination. That one basically is like just to understand what Medicare covered preventive services are available. So we call the IPPE. So that one is just available one time. the first 12 months once the patient is enrolled to Medicare Part B. Okay, next. Okay, yeah, just leave it here. So, so for the first 12 months, you can do the IPPE. So it's just one time, is the initial one. You're gonna talk about the preventive services, and that's it. The G038, G0438 is the initial annual visit. So you can do one IPPE and you can do the initial annual visit 12 months after the patient received the IPPE or 12 months after the patient was enrolled. So a lot of the times nobody does the IPPE because they don't know. But if you think about it, you can bill 2 times, like once every year for like basically two initials annual wellness visit, which is the RVU is 2.43, which is a very high RVU. So sometimes we don't know, but that's something that we can do. And then every other, and subsequently every year, then you just do your annual visit, but just the follow-up, not the initial one, which is a lower RVU. Next. Yes, so which is the G0439. So that's going to be after the initial one, so 1 year after, but you can do the IPPE 1 year right after they enroll, the G0438 1 year after the IPPE, and then the follow-up. Next. Okay, so basically the annual wellness visit is not as simple, just like, okay, let me talk about the preventive services and that's it. Actually, you need to like write and discuss with the patient a little bit more things that are required by the Medicare, like demographic data, self-assessment of health status, psychosocial risks. So you're gonna talk about, risk of falls, depression, the mood, any behavior, are you safe at home, your ADLs, IADLs, which is very important for our patients, any substance use disorder. So you're going to talk about a lot of stuff. The cognitive assessment screening, very simple here. You're just going to talk like, hey, do you have any memory issues? Do you have any family history of dementia? So here is very superficial. That's why you can do the cognitive assessment, all the workup with the test and everything on this visit. The issue is the time, how long you have to do that visit, and then on top of that, a cognitive assessment if they screen positive here. Next. Next, so this is basically the template that we created here. So makes it easier, right? So once you have a template created of all the things that you need to ask, and you basically just click, makes it very easy, even though it's a lot of information that you need to ask. A lot of things just auto-populate from your EMR, right? Like family history, past surgical history, socioeconomic history, sometimes just auto-populate for you, and you just need to ask the other few things like your ADLs, IDLs, your function ability. So it's kind of like a yes or no. So once you get familiar and used to, goes pretty quick. In the beginning, you can struggle a little bit, but after a while, you get used to with this template, it gets easier. And the AWV template is like you're going to ask us about vaccine and screening recommendations and everything that is positive, like smoking, cardiovascular risk, everything that is positive, you can build that on top if you do the right attestation, which I'm going to talk later. Next. Next. Okay, so the cognitive assessment. So they are different. So the cognitive assessment for the AWV, very simple. So you're just gonna screen, ask a few questions, and if it's positive, you can schedule the patient to come just for a cognitive assessment visit, or you can do at the same time if you have enough time. So of course, as we know, we check about the abilities, cognitive level, capacity, ADLs, IADLs, ask for collateral information from the family. And like I said, we can do in the wellness visit, but not in a standard visit. Medicare doesn't allow that because the cognitive assessment visit take over because of the RVUs. So basically what do we do on this visit? Well, we need to, if it's positive, just the questions asking for the patient, for the family, we need to use some of our tools, right? So a mini cog, MoCA, MMSC, RURAS, memory impairment screen, AD8, dementia screening. So any of those that you like. We usually use the MoCA here, even though it takes a little while. You can try with the mini cog first and see if it's positive, and if it is, you can go to the MoCA. But that's basically one of the things that you need to do for a cognitive assessment visit, not necessarily just for the little assessment on the AWV. You don't need to do that, just if you want to do a mini cog, which is faster. USPSTF, though, doesn't like recommend, you know, like usually like the isn't sufficient to recommend like the assessment for every older adult. But this is just a recommendation for USPSTF. In our AWV, we ask any patient, and if it's positive, we do the further assessment. But USPSTF doesn't think we need to screen, otherwise, just if the patient complains or the family has a concern. Next. Okay, so like I said, if you're suspecting a cognitive dysfunction, even if the patient said everything is fine, but just talking with the patient, you're like, something is wrong. I think I need to do further assessment. We do it. The cognitive assessment visit is a standalone code and is higher than a regular visit. You can co-bill with wellness visit or preventive services if they screen positive and you have enough time to discuss with them. Like I said, you cannot do with a standard visit. You cannot like split bill with a standard visit. This one takes over. And yeah, if the assessment is positive, that's what you can do. You can do at the same time, or you can do it in another day. We have like a specific clinic for that. So if my patient is screen positive, I just schedule them for another visit with us, just for the cognitive assessment visit, because it takes a long time and I don't have enough time to do on the time of the AWV or any other visit. Okay, next. Next. Yes. Yeah, like and like the other item was saying, like the total RVUs, it's higher, like a normal visit for a new patient is 2.5 and for a cognitive assessment 2.8. So like I said, that's why it's not that simple. If you ended up doing a cognitive assessment visit, you need to meet all these criteria. So like a good physical history and physical exam, medical decision making of moderate or high complexity, which is going to be if the patient has a cognitive impairment. You're going to use like our tools, right? Check for ADLs, IDLs, do medication reconciliation, check for depression, for mood, for safety, driving. Do we need to report to the DMV? Talk with family if we can, and patient allowed for collateral information, social support. So it's a lot of things that we need to address on the visit. That's why sometimes it's very hard to do with the AWV. Usually take like 15 minutes to do this visit, and like I take sometimes even longer. So that's why I don't see doing together, but some people they do, some of the providers. Next. So how can we maximize the preventive service? How can we make our life easier? Well, so a lot of these questions that we call like the health vital signs, they can be already like in a questionnaire, right? Before the patient come and see us, like smoking, alcohol, questions about all the metrics that we want to address, that we can address, or maybe that we can bill for, right? Exercising, healthy diet, weight, alcohol, like I said, STD risk. So if it's positive, so they can fill out the questionnaire before seeing you with the medical assistant. And if it's positive, when you see, that is something that you can discuss with the patient and you can bill for that. So I think that's very helpful. That's what we do in our clinic too. You just need to make sure how often can you bill for each of the preventive services. But if you don't, of course, it's so hard to memorize everything, but your office is gonna let you know, because if you bill and you're not gonna be reimbursed for that, it's because it's not due. So sometimes I just do it, and if it's due, great. If it's not, then I'll just wait whenever it's due, right? But sometimes it's good to know, but I know it's hard to memorize everything. Next. The good thing for if you do that with the AWV is that the AWV you do once a year, right? You cannot do more often than that. And the majority of these preventive services, they are also qualified once a year. So if you do them on the AWV visit, probably going to be reimbursed for all of them. So that's a good tip to have. It's just like, if I'm going to do the AWV, let me do my preventive services, because that's probably how often I'm going to be reimbursed for that. So let's try to have at least 30 minutes slot in our institution. Sometimes we have an hour for new patients for the initial one, and then 30 minutes for the follow-up. So we have a lot of time to discuss everything, to add the preventive services if they screen positive, and even address other issues that I can split view my documentation, my note. And of course, like I said, address the positive screening, and then if there is any overlapping counseling, like if it's positive for smoking, it's positive for cardiac risk, and you want to address everything, we just want to make sure you document right. Okay, and I'm going to give you a few examples for that. Next. Next. So this is a nice graph. So basically it shows the preventive services, the CPT code, the RVU, so how much you're going to be paid. This year, the RVU for cognitive assessment increased from 3.44 to 3.80. That's why I put it over there. And then how long you need to stay to talk with the patient about it. So for example, depression screening, like less than 15 minutes is fine. The smoking cessation, depending, like if you spend 3 to 10 minutes is 0.24 RVUs. If it's more than 10 minutes, is a little more. So it's like give you the time that you need to do your attestation. And of course, if you spend that time with the patient, we don't want to lie in our documentation. But if you spend that amount of time, more than that few minutes, then you can bill for that preventive service. And then here show like the recommended diagnosis and the interval. So how often can you bill for that? So you can see some of them 365 days every year, some of them 180 days. The advanced care planning actually is every time the patient change their mind. So if they are DNR, they change for full code, or vice versa, you can bill for that because it's a change of status. Next. So basically, what do we need to do to document? So it's just 3 components: the amount of time you spend counseling, the CPT code, which is going to be on your EMR, is going to populate for you, and the diagnosis, right? So one important thing is that the time documented cannot overlap between the time for the standard office visit and the AWE, and the time for the preventive services. So for example, you do your AWV, and then you're gonna, like, your depression screening was positive, you consult the patient, like you spend like maybe like 10 minutes counseling the patient, you maybe started even a medication, but you spend that time talk with the patient about the depression, then you're gonna document that. I have a few examples I'm gonna show you, but you're gonna have to document, like I spend 8 minutes, 9 minutes, 10 minutes talking with the patient about the depression screening, and this time is exclude the AWB visit. So you need to make sure you write that, and I'm going to show it to you. Next. So appropriate documentation will improve your Medicare reimbursement, right? For the services that you provide already, you're just not documenting that. Is not exclusive for primary care providers or geriatricians, can be billed for any specialist. So if somebody else billed before you and you did it and you didn't get the reimbursement, probably because somebody else already claimed for that. And if a PCP document and bills for a particular service after a subspecialist, like I said, so you just the geriatrician, the primary care provider is not going to be reimbursed. So it's very hard to know if somebody else billed before us, but you're going to know if you're not going to get reimbursed for that visit because somebody else did. So anyone can do it, any specialist, any nurse practitioner, mid-levels, anybody can do it. Next. Okay, so that's the time attestation I was talking to you guys. So for example, the advanced care planning, okay? So template suggestion. I spent 15 minutes with the patient in counseling and discussion of goals of care, code status, and advanced directives, and detailed the assessment and plan, excluding visit time and annual wellness visit time. So that's what is important. So if you do that with advanced care planning, you need to make sure that the time you spend is excluding the time you're spending for the wellness visit or the visit or just the standard office visit. And if you add more preventive services, and excluding the depression screening time, the weight counseling time, you need to make sure you exclude that you spend that 15 minutes just talking about advanced care planning. So if you didn't spend that amount of time that is required to bill, that you don't do it, but you need to make sure that time is not a combined time, but was spent specifically for that preventive service. Same thing for the depression screening. So I spent 10 minutes with the patient on screening and counseling about depression, excluding advanced care planning and wellness, annual wellness visit time. So you need to be very specific. So we just have like little template smart phrases of each one. So then it's very easy to put on your note if that's something that you addressed. Next. Conclusion. So the role of the primary care physician is to provide comprehensive care, right? So that's what we want to do for any patient, and for our older adult patients is even more because of all the comorbidities, polypharmacy and everything. If you don't document what you're doing, it's not going to reflect all the care that you provided. So pre-visit screening questions is very important. So that's when is the time that you see like, okay, which screening can I address with my patient? So they did their homework already. And it's going to be easier and helpful to see like which items you can cover during that time and do you want to cover also. Because sometimes you cover the last visit or you're going to cover on the next visit because you don't have time. So that's very helpful. Increasing the time slots. I know sometimes it doesn't depend on you, it's on your practice, but if we have more time for the AWV visits, that's going to be very helpful to just add more preventive services if the patient screen positive. And having templates. I have templates for everything, for my AWV, for all my time attestations, because it makes your life easier and makes everything like faster and more effective. Next. These are my references. Next. Next. And thank you. If you guys have any questions, I'm happy to answer them.
Thank you, Dr. Ekler-Jaguar. Is anyone having any questions? Okay, thank you once again, Dr. Ekler. Now we came to end of our day 2 webinar. I would like to thank all the participants and attendees of today's webinar. Hope you enjoyed the webinar and gained knowledge from the sessions, and request you to attend the tomorrow sessions too. If you have any queries, you can mail to the concerned program manager. Thank you very much. Hope to see you tomorrow. Bye-bye. Have a nice day.